Hello, everyone, and welcome to week 4 of the 18th Annual Symposium on the Conservation of Tortoises and Freshwater Turtles. Thank you for joining us. I just want to give everyone a reminder that you have 1 week left to view and interact with poster session number 1. September 3rd, poster session number 2 will be up, and you'll have a whole new slate of things to look at. Today's sessions are titled Field Techniques and Turtles on the Move, and they come to you live from around the world. And Vicki Hudson from South Africa is here to kick things off for us. Vicki, are you ready?
Yes, hello.
Hello, Vicki. Enjoy, everyone, and see you later.
Hi, everybody, from the southernmost tip of Africa. I'm just trying to, for some reason, the chat box doesn't want to come out now. I'll give it a little bit of time. David, can you see my screen?
I sure can. It looks great.
Thanks. I'm not sure why the chat box has now decided not to.
Not a problem. We can work with it.
Okay, so I'm going to start. Hi, everybody. Yes, I'm coming to you on behalf of Jim and Ross, my co-presenters from almost the southernmost tip of Africa. And lots to say, very little time, so I'm going to jump right into it. Today, I'm going to discuss the use of conservation detection dogs in a Mark-recapture program for the geometric tortoise in the Western Cape. The outline of my presentation, I'm going to briefly introduce the geometric tortoise, discuss its major threats, conservation challenges, and illustrate this with a case study. I'll introduce our Mark-recapture program, briefly describe the study area methodology, discuss the preliminary results, and then just touch on a few added bonuses we found with the program and close with acknowledgements. So the geometric tortoise is commonly known as the Fabergé egg of the Chelonian world. Its conservation status has gone from vulnerable to endangered, and in 2015 it's classified as critically endangered. It's also been included in the 100 most threatened animals, plants, and fungi by members of the IUCN Species Survival Commission. I've lumped the 3 major threat categories into habitat loss, fragmentation, and by alteration. By alteration, I mean a change in perhaps the grazing techniques, or also the spread of invasive alien plant vegetation into the habitat. Then the second threat is unplanned and frequent fires. So the geometric tortoise does occur in a fynbos vegetation type, which is occurs in a fire-driven ecosystem. However, recently our fires have become much larger in extent and more frequent, mainly due to anthropomorphic reasons. And we don't have because of habitat fragmentation, we haven't got the surrounding population of unburnt natural felt with geometrics that can then repopulate the burnt areas and account for the mortality. And then lastly, something that's come on the radar recently in the past 5 years is climate change. It's happening at a rapid rate. We're seeing the results on the ground, and we're just wondering if the geometric tortoise can adapt to the pace at which the climate is changing and the effect it's having on the vegetation. So the conservation challenges, despite its visually prominent and contrasting patterning, the cryptic coloration of the geometric tortoise actually makes it very well camouflaged and difficult to find. It's one of the most sedentary tortoises, so your chances of finding it out in the open, walking around all times of the day, are very slim. Also, humans only use one sense to find tortoises, so that's a visual sense, and they experience bias in terms of location, size, and search consistency. So just an example of this, in my top right-hand corner, you'll see a picture of a stand of eucalyptus trees, which are invasive alien vegetation in South Africa, and a dog hidden right in the middle. This occurred during a search and rescue that we were doing, we would never have searched the area because we just assumed that the geometrics occur in fynbos, natural vegetation, and very short shrubby vegetation. And the dog pulled us into this area and found geometrics eking out a survival here. And we were fortunately able to remove them and save them and put them in the protected area, the reserve. At the bottom, yes, I know it's not a geometric hatchling. It's a common puddler hatchling that they co-occur with the geometrics, but just to give you an idea of the size range that we find and that the dog finds. And for the dog using a scent, it's not about the size. The bigger the size, the more scent the tortoise produces. We found that with hatchlings, they actually move a lot in a very small area and create quite a concentrated search scent for the dog, or search pad almost for the dog to pick up on fairly easily, as opposed to a larger adult that is not moving around for a long period of time, is holed up in a bush and with very little air movement, will be more difficult for the dog to find, even though it's larger in size. The other thing, we don't have the luxury anymore of being able to have teams of 20 to 30 people for 3-4 days a week at a time out in the field, combing every single bush to see if there's a geometric underneath it. It's just not possible anymore with the constraints we have. And we've seen the results. We miss a lot of tortoises with that. Also, we don't have the same searchers every day in each team. It's different people all the time. So we have no consistency in terms of skills or in terms of surveyors. The closest we've come to doing a comparison between the two search techniques occurred when we had a development site with 6 hectares split in two by a road, and the developers put a tortoise-proof fence around it. And the human team came in and searched the area and removed what they thought was almost 99% of the tortoises. That was 28. They were removed from the area. I was then asked to come in with the dogs and do a final sweep to pick up any 1 or 2 that may have been left behind. As you can see, at the end of the day, the result was that the human team had 1 tortoise per 6.4 hours search time and 0.7 tortoises per searcher. And then for the dog team, it was 1 tortoise per 1 hour search time and 12 tortoises per searcher. So as you can see, the biggest difference occurred in the cost per unit effort per hectare. The human team in blue cost considerably more than the dog team in red. And then in terms of number of tortoises found per searcher, the dog team in red did a very lot better than the human team in blue. Obviously, this was not an exact comparison because there weren't the same amount of targets for each team to find, but it's as close as we have got to comparing the two to date. So the Mark-recapture program. In 2015, the Turtle Conservancy secured ownership of 365 hectares of the last stronghold of the geometric tortoise. Cape Nature and the Turtle Conservancy formed a partnership and initiated Mark-recapture surveys using conservation detection dogs in order to find out more about the population at the site and to inform conservation management of the species on the ground. So the study area, at the moment, it doesn't extend across the whole 365 for this Mark-recapture study. We've concentrated on 2 blocks, northern block and a southern block, divided by a highway. The northern block is further divided into 4 blocks and the southern side into 2. And then what I try and do is 2 transect loops of 2 hours duration each for a search. So that's 4 hours search period per day. But they are the ultimate goal is to find as many tortoises as possible in the time permitted. We only search in spring and autumn. We don't do any searches in summer because it's detrimental to the tortoises, and also there's the threat of snakes to the dogs. So yes, this is an example of a real life, what the transect looks like on the ground. It doesn't always go to plan. It's not perfect, but that's just how life is, because there are, as I said, abiotic and biotic factors that affect it. So the purple track is the dog track, the white track is the human track, and the yellow dots, the yellow points are the geometric finds, and the blue points are the angulate finds. As you can see, the dog pulled me right off track to find geometric male number 108. It was out in the open, and I suspect that on that day, if there was a breeze blowing from the southeast with the tortoise out moving about in the open, the scent would have been able to be picked up from quite a far way away, and the dog just pulled me right there to that tortoise, which was off the transect loop, but I have to trust her and listen to her, and at the end of the day, we got the find. So now we come to the part of the presentation that I don't have anything to do with. I'm just the dog lady that finds the tortoises, captures the data, and sends it off. Ross Keister is a statistician from the Turtle Conservancy and scientist, and he works out the estimated number of tortoises using the Cormack-Jolly-Sieber method in R-recapture. N equals 542, where N is the total number of different individuals that were marked and possibly recaptured according to R-recapture. As you can see here, there was a distinct drop between 2017 and 2018. So of those 542 that were marked, we've had 98 that were recaptured once and 11 that were recaptured twice or more times. For example, number 2000 was marked and had 3 recaptures subsequent to that. And number 6000 was marked in 2015, recaptured in 2016, and unfortunately died in 2019. We've also found more males than females. Discuss that later. I know it doesn't add up to 542, but some records were removed in finer analysis. We use the Honegger notch marking system, notch marking with a file or a Dremel tool. And yes, when there's not many tortoises at a site, I can work alone. But at this site, there are a lot of tortoises, and we try and handle them as little as possible and in the shortest amount of time. So what Jim and Ross have been instrumental is helping us train up field rangers. This is Rudy and Winnie. Rudy is a star. He can notch mark. He follows the numbering system. He takes all the measurements, and Winnie is there to record everything and capture it electronically. So in the discussion at this stage, we can say we have a population of roughly 1,000 tortoises, but we do need more data for improved accuracy. Yes, we found more males than females, and this I attribute to behavioral characteristics, especially in the spring. That's mating season, and I often find more than one male in a certain area. It can be 3 or almost 4 in a wide radius, which I don't find with the females. So I suspect it's got something to do with their behavioral traits. As I alluded to, there was a definite decline in 2017, and we attribute this to the drought and to a fire on the southern side. The drought, we suspect that the tortoises, perhaps we haven't picked up many mortalities directly as a result of this, well, yeah, in the field. But what I suspect is that they've moved into a much more densely vegetated support area to the west of the study area, which is much more sheltered. And I think with the drier conditions, they've moved in there. We did have a fire in the southern side, which is a smaller section, and this did account for mortalities. Getting back to climate change, which I raised as one of the recent threats. As you can see in this graph, over the past 5 years, we've had significantly less rainfall compared to the previous 15 years, and this is really a concern for us going forward. And then just to touch on some added bonuses. So unlike in Africa, we've got the elephants, the rhinos, the lions, the leopards, the wild dogs. They're all very sexy species, and people can come out and see them, relate to them. They're cute and furry, and the geometric tortoise just doesn't have the same appeal amongst the public. It's also very difficult to garner support and tell people about its species or a little animal that they can't come out and see. We don't reveal the location where these tortoises are found in a very specific area in the Western Cape. We don't have volunteers coming out to do our surveys. So it's very difficult for people to know what we're actually talking about. When we started with the dog project, we just put a small article on our website. One media person found it and did an article, and since then the media awareness has just exploded. We've had many stories written, we've had lovely TV programs done, and the combination of the dogs and the geometrics and the story that goes together with it has really been popular. And we've increased people's awareness of it. People are talking about it, especially in the Western Cape. And it's been a real bonus because people understand the importance that small species have. I've also been fortunate in able to show this technique. It's a low-tech, non-invasive survey technique in the conservation world to conservation students, you know, both from our Technicon up and coming conservationists in the next generation, as well as international students. So this is quite a unique opportunity. I've also been able to share with colleagues from or friends from the Durrell Institute in Madagascar. They've come over and seen how we use dogs to find tortoises, and we've also had the opportunity to go over to them and explain to them, look at their circumstances and see how they could possibly use dogs. The dogs are not only used in Mark-recapture programs. As I said, they're very effective in search and rescues, both from fire and development, just in terms of being able to respond rapidly and being able to go at different times. And yes, we've met some rich and famous and supermodel people along the way, which has been fabulous. In closing, I'd just like to acknowledge Jim Juvic, because without him, the pieces of the puzzle would never have come together. So thank you, Jim, respect, and congratulations on winning the Baylor Award. You really deserve it. I'd like to dedicate this presentation to Professor Emeritus Margarete Hofmeyr, who passed away at the beginning of this year. Rita was more than a mentor. She guided and supported us on every step of the journey, and she's going to be sorely missed. So thank you, Rita. That's all I have to say for now. I hope I'm in time. And yes, let me try and open my chat box.
And if you can't manage, I can just give you questions here. We're going to wait just a little bit to see if we have some come in. And after we wait for questions, up next is going to be Patrick Kane telling us about utilizing do-it-yourself open source technologies to make life easier on the turtle biologist. Vicky, question number one. Someone says they missed the first few minutes, so they apologize if you already discussed this, but are there other tortoise species in the area you're working in? Will the dogs only find geometric tortoises, or could they adapt to any tortoise study?
Oh, that's an excellent question. And I was actually going to include it, and then I cut it out of my talk. So I'm so glad you asked that. It's one of the favorite questions, and it's something that we didn't know before we started the project. So yes, we have two other tortoise species that occur with the geometric. So it's the common putlooper, and then your angular tortoise, or we call it the Roipens chasina. And each of those species actually has a different scent. So if you wanted to, when we do surveys, it's very important for us to know the ratio of geometrics, angulates, and putloopers in an habitat, because it's very important. I won't go into detail now. So we need to find all those three species. So the dogs are trained to alert on all those three species. However, if you wanted to just train it on geometrics and exclude the other two species, there's enough of a distinction in the smell between the species that you would be able to do that. So thanks for asking one of my favorite questions, and something we didn't know going in, and something we found out with training. So thank you for asking that.
Second question comes from Samrita Padma. Do dogs harm the animals at all?
So it's very important with the conservation detection dogs that they have a passive alert. So the dogs don't interact with the target at all. Once they locate the target, the dog will either do a sit or a down next to it. So it does not pick it up. It doesn't carry it to me. It doesn't scratch the tortoise. And this is a very strict rule, and we enforce it in the training, because otherwise we wouldn't have been able to work in protected areas if the dogs had any negative effect on the target. So yeah, we're very strict about that. And we also, that's one of the reasons we don't do surveys in summer, is because the tortoises excrete, you know, when they are disturbed, even if they aren't touched. And that loss of liquid, because we're in a winter rainfall area, is detrimental, and they can dehydrate. So we make sure that the tortoises do not harm, the dogs do not harm the tortoises in any way.
Fascinating. Vicky, one more question. You mentioned biases in human surveys. Are there biases when using dogs to survey?
Yes, I think with the dogs, so if the wind is blowing very strongly, or it's blowing in turbulent, through the vegetation in turbulent circles, the dog is going to find it very difficult to find the tortoise. So we get very strong gale force winds, and that takes the scent further away from the target much quicker. So the dog battles to find to actually pinpoint the target. So yes, there are factors that you have to take into consideration, and not all conditions are suitable to use dogs in. I'm trying to think offhand of other biases with dogs. They do use more than one scent. Yeah, there are conditions that you can't use dogs in. But yeah, apart from that, I must actually go and think about that a little more.
Okay, I've got another one for you. What is the budget generally to train a dog and a handler for this type of research?
Okay, so it depends on whether the dog is already trained in the game of detection work, because all detection, whether it's your drug detection, explosive detection, starts with the basic principles. So the dog is finding a tortoise, not because it loves tortoises. I think that's an important principle, but because it wants a ball reward. That's its paycheck. So if the dog already knows that, and it's trained to that level, you could probably buy a dog. I'm trying to think now, in our rands, or you're probably looking at anything between $1,000 and $10,000 to buy a dog like that, and then to have it trained specifically for your type of target. But the dog is also only as good. It's definitely teamwork. So then you've got to have the handler. So the costs vary. It's much easier if you do it in-house. But again, yeah, the dog and the handler dog team is doing 4 to 5 times the work of a team of 10. So yeah, it's difficult for me to relate costs here from South Africa, but it depends on various factors. Yeah, the initial outlay, oh, that's the other thing, the initial outlay might be expensive, but then for a conservation detection dog like this, my dog is going on 8.5, and she's still working. So I've had 7.5 years work out of her for my initial outlay.
And it's great. And we have one final one for you from someone who joined late. Do the dogs have to start training as puppies, or can an adult dog be trained to do this as well?
Yes, an adult dog can definitely be trained to do it. And with puppies, we definitely don't start with a live target because they're just too interested in playing with it. So it's normally up until a dog is about 2 years is probably the maximum we'll take it for an adult dog to train it. The most important thing is the dog interested in a ball and a reward, and is it motivated to do it and to use its nose? So yes, adult dogs can definitely be trained for this.
Great. Thank you, Vicki. It's always a fascinating subject. And that was the final question. And now Patrick is up, and we're going to let him take over the screen.
All right. Yeah, so hello, everybody. I'd just like to start off by giving a big shout out to Christina Jones. She set up and organized our GPS data logger workshop for basically every single turtle meeting in 2020. And I was so excited for that I had actually forgotten that I submitted this abstract to TSA last year. So if you're in Tucson, stick around. There's some updates and changes. But if you haven't seen this, or if you weren't in Tucson last year, the thing I like people to take home from this talk is that you don't always have to be limited, or your research questions don't always have to be limited to what's commercially available. So commercially available. So any technology that already exists. So for example, I work with these guys. These are Scalopus lizards, fence lizards. The males are highly territorial. And so if you have a resident male and an intruder male comes in, they'll do this stereotyped behavior where they'll square off with each other laterally and do push-ups. And so they evaluate each other's fighting ability through these push-ups. And if they're evenly matched, then they might engage in physical combat. But usually disputes are settled just by doing these push-ups. And it's a fascinating behavior, and Scalopus lizards do it, and all kinds of other genera and lizards do it. And so there's been a lot of focus on what information they're conveying to each other through this. And then one of the good ways to do that or study that is to build a robotic lizard that can mimic the push-ups, and you can manipulate it and see what's going on. Now, I'm less interested in what the lizards are talking to or telling each other about their fighting abilities. I'm more interested in predation. And so a lot of the robots that were built for the conspecific behavior, the lizards talking to each other, are super expensive. And I needed something much cheaper so that I can put lots of them out in the field and try to elicit a predation event. And so I just went ahead and built my own robotic lizard. And so you can see the inside of it, or the outside of it, just a latex lizard. There's a servo motor up here in the red cup that pushes a paper clip up and down, so it moves the latex lizard up and down. And so it's also much, much cheaper than what was available. So now I can make lots of these. You can see it in action here. So this is up at the Toledo Zoo. I know it's winter. Lizards aren't out in winter, but this Australian magpie didn't seem to matter, or didn't seem to mind. And so, yeah, now I have a robotic lizard that can elicit predation responses. And one of the other cool things are that I added an accelerometer to this robot. So an accelerometer is just a component that you can that are actually on all of your smartphones. So it detects the orientation of your phone, so it can change the screen. But in this case, I utilized the accelerometer to detect when the model gets jostled by a predator. So it'll detect that motion. It'll use the real-time clock to create a timestamp, and I know exactly how long that model has been out in the field before it gets attacked. Now, I'm not an electrical engineer. I'm not a computer programmer, but I was able to utilize this this device called Arduino to do that. So you can see the Arduino is this blue chip in the middle, and Arduino is meant to get non-electrical engineers, non-computer programmers into making stuff. So it really makes it easy for people to jump into it and create something without necessarily having to have several degrees in electrical engineering to do it. And so Arduino is basically two parts. You have a hardware part. So the Arduino is a microcontroller. You could think of microcontrollers as a computer, but it doesn't have anywhere near the processing power that a computer has. And so it can do very basic instructions very, very, very well. And so there's lots of different flavors of Arduino. So you have the flagship one is the Uno. If you want something smaller, you can use the Micro. I use something even smaller than that called the Pro Mini. If you need something that has lots of processing power or lots of power, you can go up to a Mega. But all of these devices, all of these things are based off of a microcontroller chip called a AT Mega. In the bottom right, you can see a pinout of that chip. And in the top right, you can see the chip in the bottom left on that. And so these boards are just breakout boards. It makes it easier to access all the pins on these microcontroller chips. But so that's the hardware. That's what you use, and you program and connect all kinds of peripherals or devices or LEDs or whatever you want to. The second part is the software. And so programming in machine language is incredibly complex and difficult. And so the folks at Arduino kind of take that C++ based machine language and put a user-friendly, happier shell on it. So it makes it, a lot of the functions are intuitive and easy to understand. It makes programming a lot easier. So yeah, that's the two components to it. So if you have the hardware and the software, you could do all sorts of stuff. And all of this is in the open source community. So when somebody uses this to make a project, they put out the schematics and the software so that anybody can access it and either replicate it or manipulate it to what they want. That's basically what I did with my robotic lizard. Is I found plans online and just changed it to what worked for me. So there's all kinds of cool projects. Like one of my favorites is somebody made a Twitter account for their plant and put a hygrometer into the plant. So when the plant starts to get dry, it'll tweet, "Hey, I'm thirsty. Give me some more water." Or on the bottom right over here is somebody who sewed a bunch of LEDs into their jacket so that they could have a turn signal for when they're riding their bike in New York City, and probably be more obvious to motorists. Right. So while I was working on my robotic lizard, my colleague who's working on box turtles complained incessantly about how he would love to get a GPS data logger on his turtles, but on a shoestring graduate student budget, he couldn't afford them because a lot of these are pretty expensive. This was a couple of years ago even, and they're still pretty expensive, several $100s to into the thousands of dollars. And so if you want one of these, usually you can only afford a couple of them, and if you lose them, that's a really big deal, which they tend to disappear in the field. So just working with what I knew about Arduino on the robotic lizard, I did a cursory Google search on GPS data loggers and found all sorts of stuff. So lots of people have built these data loggers, and so all I needed to do was just figure out the right components. So I found a bunch of GPS receivers that I could possibly use, and use some of the code and change things to where I actually built a working prototype. And this looks hideous, it's ugly, but it was a proof of concept, and it worked. I even got into animal testing fairly quickly, and yeah. So over the years, we've kind of refined the design. We put out a paper a while ago that introduces a lot of these things. It gives you schematics and a walkthrough. There's a video to show you how to build these. With our data logger workshop or GPS workshops, we teach people how to build one version. That's this is A in the bottom right figure. That's a lot easier, bigger components. It's easier to build. But the more kind of sophisticated, more data points available and that sort of thing is in the bottom right. It's a little more difficult to put together, but yeah. So we've refined it and put that out there. So the code is out there, the design is out there, and yeah, if you want to put GPS loggers on turtles, then this would be a good starting point. People have picked up on this. So there's a paper that came out a couple months ago where they started using these on ringneck pheasants in South Dakota. So they modified it a little bit, and yeah, and they found some success with these. This would be a good paper to go look for too if you want to look at all the other stuff that you would need. So soldering iron, solder, things like that. We don't talk about that in our paper, but this one does an exhaustive equipment list in that paper. I've also started working with someone at the University of Georgia, which is right down the street. He does this sort of thing with pigeons. So he makes a hat for pigeons. But the paper that they put out 2 years ago had very large components, and they were worried that the package was weighing down and inhibiting pigeon flight, because they want to look at just basically where pigeons go, how they choose different destinations, and how to get to those destinations. And so with the accelerometer I use in my robotic lizard, I can then put that into the GPS logger, and it's much less of a hindrance to the pigeons. And with the accelerometer, you can actually get wing beat patterns. So you can look at every time the pigeon flaps its wings and how frequently, and you could do stress and energy usage, and all sorts of cool questions can be answered with those data. Now, with our GPS loggers, you still need to collect the data. So you need to find the animal again so that you can download the data. And the classic way to do that is with VHF transmitters. And the DIY world is definitely into that. So folks who build model rockets build their own VHF transmitters. So they have a lot of articles on how to build them. So that when you shoot the rocket off, you can use radio telemetry to go and collect your rocket. There have been a couple of engineering type folks who try to make the smallest package of VHF transmitters, but they do publish their schematics, and schematics are a whole different ball of wax when it comes to learning and understanding how those things are. But it is very approachable. It just takes some time and look at a lot of schematics, and you can then take that schematic and then put parts together. So I've been talking to somebody in Virginia who builds their own VHF transmitters for box turtles specifically. And so he's not an electrical engineer. I think he's a retired uh, resource manager, park service guy. So just in his retirement, he likes to track box turtles. So he used these designs and built a fairly large VHF transmitter. But with I know about the GPS logger and the smaller surface mount components, I'm going to take his design and try to make that a much smaller, more efficient VHF transmitter. So um, these ones, at least with the components he uses, are actually just a couple of dollars worth of components. So if you compare that to some commercial ones, they're 80, 90, over $100. This could be a cost-effective way to introduce VHF to a project. But with VHF transmitters, you also need an antenna, and Yagi antennas are probably the most popular antennas out there, and they could be a little costly, 80, 90, over $100, or they're like 200. I guess I don't really know that, but they can be costly, or at least more costly than these VHF antennas. And the ham radio world is a huge DIY community, and they build their own Yagi antennas out of tape measure, so metal tape measure and PVC pipe. And so this is one that I put together. There's actually an equation you can use to figure out the element lengths based on what frequency you're going to track to make it more efficient. And this is just a couple of bucks, if that, if you don't already have PVC pipe and a metal measuring tape there. So yeah, if you back over one of these with your truck, or your field tech leaves it out in the field, it's not that big of a deal. Okay, so VHF transmitter, antenna. The last big costly thing of radio telemetry is the receiver. And for the receiver, I would focus on something called software-defined radio. So where typical radio receivers use a crystal to interpret radio signals, this uses software. And so you get two things from this. One, you get the audio that you normally get from a VHF receiver, the beeps, but it can also translate that into a spectrum, so a visual component to it. And this software-defined radio is literally just a $20 USB dongle that you could plug into a computer, or there's another thing you could use if you want to make this more portable. It's something called a Raspberry Pi, which, unlike the Arduino, it is actually a computer, so it has lots of processing power. So you could put a little LCD screen on this and plug in a USB dongle, attach your measuring tape Yagi, and you've got a full system that's super cheap compared to what's commercially available. And because it has this visual spectrum component to it, you can actually find signals just by looking at the spectrum. So I literally took a bunch of VHF transmitters, dumped them on my bench table, and I was able to figure out what all of the frequencies are just by looking at the blips on the spectral output of this. And that does probably mean that I could go out to somebody's field and find all of their animals without knowing any of their frequencies. So that is something to keep in mind. This is kind of new, but this is, I guess, for conservation purposes, it's definitely something we need to start thinking about. It's not, yeah, anyway, so we can talk about that later. Well, so just some other odds and ends types things. So I've been working on building environmental data loggers, which is a general purpose, temperature, moisture, humidity, all sorts of that sort of those kinds of things. It's much cheaper than what you get out there as what's commercially available. Another thing that we're about to put to the test is a nest activity logger. So this is I just combined the real-time clock from my robot, the accelerometer, and the same memory chip, and you could put this into a turtle nest and then cover it back up, and just whenever the turtles hatch, they'll start to move around and they'll jostle this logger, and so you can get nest activity data. So when they start to hatch and when they move around, how much they move around. And I guess if the movement stops, you can also figure out when they leave the nest as well, without you ever having, I guess only having been there the one time. So this is just a prototype. I know it's really big and clunky, but we're just trying to get a proof of concept to start putting in. Well, I guess it's kind of late in the season, but to start seeing if it's feasible. And then one of my new favorite things is photogrammetry. So I have a student working with antlions, and antlions in the larval stage build these pits that they sit at the bottom of, and something will come along and fall in and they'll eat the prey. So they build these pit traps. And one of the classic things to do with antlion larvae is to look at the pit morphology. So you can look at pit size, pit depth, um, density of pits, and try to figure out if those are correlated with or influenced by feeding, hunger, whatever, species diversity. But in a I found that it was very difficult to measure pit depth, because you can't put a ruler in it because you'll mess up the pit. One of the papers I read, I contacted the author and he said he put string down in it until it touched the bottom, and then you pinch it off with your fingers and measure the distance of the string. And for the life of me, I could not figure out how it worked because you put the string in and then my fat fingers were not super accurate, and then you can only measure one because you're lying on the ground trying to level off where it was. I could not figure it out until I found photogrammetry. And photogrammetry is just where you take a whole bunch of images from different angles, and the software will stitch those images together and make a 3D object, 3D digital object. And so this is a bunch of antlion pits. That's my business card that I used for scale. And I created a 3D digital image of that. And so you can take that 3D image and actually just with some basic trigonometry. So you take the diameter of that. There was a paper published that showed that the angle of the pit is always 45 degrees. And so it's the depth is a right angle with the radius of the diameter of the pit, and just do some trig and you can get the pit depth. And so I use this for antlions, but you could use this for turtle nest morphology, habitat characteristics. You can take digital images of um, the habitat around a nest and look how it changes over the years. A lot of people are putting these on drones, so you can take lots of very large aerial images of habitat and look at habitat loss. Or I think the most recent one, or something I saw was just the effect of a hurricane that came in and destroyed a whole bunch of stuff. So they had a before image of the habitat and an after, and you can go from tree to tree to leaf and learn all kinds of things there. So for any of the students out there, if you're going to grad school, having just even a basic knowledge of this could make you more marketable. And for those of you who are already out there doing research, I mean, try not to be limited by what's commercially available. So I guess in the comments, if you are familiar with Arduino and you've built something, go ahead, put some project you put in there that you think is your favorite. Or if you've never seen this before, what's something in your research that you could use that you can't seem to find that's commercially available. So yeah, I guess with that, yeah, that's all I have. Thanks for watching. And if you have any questions, yeah, I think there are a few questions. All right, have we found out habits of tortoise? Oh wait, that's from the last. Oh no, that was it. Okay. Have we found out habits of tortoises and turtles with GPS that we never thought would happen, or is this still ongoing? Let's see. I don't know anything specific off the top of my head. So one of the things I know, my colleague Matt Cross, who I talked about earlier, and I are really interested in putting these GPS loggers on turtles that, or putting them on so that they're active at night, because we don't really know what turtles do at night. Maybe they move around if it's warm or not, or whatever, because it's hard to get out and do radio telemetry of turtles at night. So I think what turtles do at night is still fairly open. And also with these loggers, we're really interested in what turtles do during fire. So you can put these loggers on turtles and take a very frequent location, like say every minute, and do a prescribed burn and figure out to whatever the accuracy is, to 2-meter accuracy, what the turtle is doing for a fire. So does it try to run? Does it hunker down immediately or something like that? And because these are fairly cheap GPS loggers, the turtle will probably survive, but even if it gets hot enough to damage the components of the GPS logger, it's yeah, it's not going to break the bank. So no, there's lots of questions that could potentially be answered. And you can also put all kinds of other components on these loggers. So if you want to put a temperature sensor um, or whatever else you want, they're highly customizable, which makes them very more attractive than commercial units that are less customizable. Okay, question 2. What if you want to examine movement below GPS resolution? Is there a workaround you've found? So below GPS, so that would be like microhabitat stuff. The best you could get for GPS is about 2.5 meters. If you wanted to step up more accuracy there, you're talking about much larger pieces of equipment, and that is definitely into the thousands. So if you want to get into like the centimeter accuracy, um, I don't think you could build something yet that you could attach to the turtle and leave. So below GPS resolution, it would be, or at least general GPS resolution, what might be difficult. I hope I answered that. Question 3. What is the battery life like on the GPS units? Would they work for semi-aquatic species? So the battery life depends highly on how frequently you take a point, and also how long the receiver has to basically listen for satellites. So if you're like in South Dakota with the pheasants, they would put them out and they would get a highly accurate fix in like 30 seconds because it's the plains, it's wide open, there's no obstruction of the signals. If you're in a dense forest, you might want to keep the receiver on for 2 minutes, which is going to drain battery power. So it is a trade-off. I have an Excel calculator that'll help you figure out if you do so many points per day and how long you open it, it'll tell you how long the unit should last. But if you do some math, you should figure it out. If you take 1 point per day and you stay on for 2 minutes, I think it'll last like 5 or 6 months, if that gives you a ballpark. And then the trick with aquatic species is that radio doesn't penetrate water very well. So if you have a turtle underwater, it's not going to get a GPS location. So I actually have this GIF here of a water sensor that I threw together. And so you could incorporate this into your GPS logger, and if it detects water, just say, don't even try to wake up because you're not going to get a signal. So you'll save lots of battery power that way. But if that turtle is basking and the GPS logger wakes up and it doesn't detect any water, then go ahead and take a GPS point. But for most super aquatic turtles, you're probably not going to get many GPS points because it's just the signal doesn't go into water. You might want to think about sonic type loggers. How effectively can we use these for undulating terrains of mountains? I don't know that I understand what that means. Undulating terrains of mountains. So like if you're in a valley or something, do you get a good signal? I think that's what that question means. Yeah, I mean, the GPS loggers are just as limited as any other commercially available one. They all have the same kind of limitations in that technology. I don't know how to deal with that. That's a good question. If you had the budget to buy these commercially, would you still go with these DIY options? Oh yeah, if you can buy something and afford something from an actual licensed engineer, that's probably the way to go. Plus, they have customer support, and they'll probably, if there's a flaw or something wrong with your unit, they'll replace it for whatever. So yeah, if you can afford it, absolutely. But what makes these kind of DIY things attractive are that you can produce a whole bunch of these for very cheap. So you can ask so many different types of questions where costs would be prohibitive. Which of the items you described do you think is the most worth trying DIY? So if you haven't been in Arduino or anything like that, there's lots of starter kits. If you look at our paper, there's a vendor called SparkFun. They have lots of guides and they explain lots of things. So I'd get an Arduino starter kit, just get something together, blink an LED, and just start there, learn the code, find people's code, read it, try to understand what it means and that sort of thing. Yeah. That's probably the best way to go. And there's lots of books, and so books that'll introduce Arduino. There's books that'll have just a whole bunch of projects that you can follow through step by step, and yeah, just learn as you go. So I think that's all the questions. All right, thank you very much.
Hello, good morning, everyone. Good afternoon. It's a pleasure for me to be here and to present my work about the diet of the pond turtle, because I could not afford to travel to the US, and I think it's not really eco-friendly to take the plane for a week symposium. So it's a pleasure for me to be here, and I will present you a part of my PhD thesis. So first of all, the European pond turtle is the only native freshwater turtle of Switzerland. And there are two native subspecies in my country, which are Emys orbicularis orbicularis and Emys orbicularis hellenica. The statue and the causes of regression. So it's one of the most threatened reptile in Switzerland. It's actually ranked endangered. And as always, it's due to human activities. The species was consumed in large quantities in the last century, and also because of the habitat loss and destruction for urbanization, agriculture, and so on. The Emys project, to go on a more positive note, I can't talk about the Emys project without talking to my turtle center. And the turtle center is located in Switzerland. Oh, sorry. And here is our facility. It's quite new. We just moved there in 2008. And we are all volunteers there. We are active since 1994 now. And the first goal of the center was to rescue the turtle, because a lot of people release exotic individuals into the wild, and it could be really harmless for the Swiss biodiversity. So we take care of all these turtles. We have now more than 2,400 turtles at the center. And we are only open on Saturday morning. So here are some information if you want to go to have a look on our Facebook page. And in 1999, we launched the Emys project with the aim to reintroduce this species into the wild. And between 2010 and 2020, we released almost 120 individuals. But the question raised to determine or to know if this reintroduced species could be a threat for the endangered species. So if this species will feed on threatened amphibians, for example. So I decide as a part of my PhD to study the diet to determine if the European pond species is really a threat for other endangered species. So to do so, we have to study the diet as a function of time. So during the whole activity period, we did a capture session to collect feces, and we did field work from April to September. We choose to collect feces because it's a non-invasive method to get the data. And with this analysis, we could also determine the difference in diet between male and females, between adult and juveniles, and determine if there is a shift in the diet during the year. So usually, if you look at the literature, the diet of the pond turtle was studied using feces by direct observation or using a microscope, but there is a huge loss of information by doing so. So for example, if the pond turtle eats a worm, such as here, it's unlikely that you find any pieces of the worm in the feces. So we decide to develop a new method of global DNA amplification and metabarcoding using PCR primer to determine precisely the diet. So the fieldwork, this is the net we use for the fieldwork. They work very well without baits. And the only thing you have to do when you do fieldwork, the weather has to be really nice because the turtles are moving a lot when it's sunny. But if it's a rainy week, you won't catch any turtles. So we spend a week each month during the activity period, setting up the net on Monday and going back to the field each day to collect the turtle. We put them in individual containers, and the day after we collect the sample. We did not keep the turtle more than a day, so we release them the next day. We did some measurement. We took the sample to go directly to the lab to extract the DNA, and then we put back the turtle into the wild at the exact same place where we captured them. So in total, I collected 174 faces from 142 different individuals. So after the sampling, the lab procedure is composed of the grinding of the sample with the liquid nitrogen, the total DNA extraction of the sample. Then you have to do a lot of PCR tests to select a PCR primer targeting the plant, the vertebrates, and the invertebrates that could be found in the diet. Then you amplify all the sample and you create the library for sequencing, and we use the Illumina Mini 6 to sequence all our data. And ultimately, you have to do to use bioinformatic pipeline to get the data and finally get the EMIS diet. So the strength of the method, what we can say, it's that we get a great amplicon lens, and with that we could precisely determine the diet of the pond turtle to the species level. What we can say, our method is only qualitative, not quantitative, so it was not possible to determine the amount of food that was eaten. And unfortunately, nowadays with metabarcoding method, it's not possible to determine the status of the prey. So we don't know if the prey was an adult, a juvenile, a larvae, or an egg, or if the animal was dead or alive. For the result, almost 90% of the prey were identified to the species level. This was really good because our method, we were very happy with the method we developed, and we identified more than 1,000 prey from 270 different species. So here are some of the invertebrate species we found. We found as well as aquatic invertebrates and terrestrial vertebrates. Maybe the pond turtle is going on land to feed, or maybe the animal just fall in the water and die in the water. We found also some vertebrates in the diet and a lot of different of plant species. So in total, on the 174 samples analyzed during the whole activity period, 90% of them contain plant DNA, 21% contain invertebrates DNA, and only 26% contain vertebrates. So to go more in detail, to determine the difference in diet by sex between the adult and the female, if you look at the metabarcoding analyze show us that 97% of the fecal sample collect on female contain plants, and 100% of the feces collect on male contain plants. The difference was not significantly different. We find the same for the invertebrate consumption. There were no significant difference between male and female, and the same for vertebrates, with a slightly higher number of vertebrates found in the male. So no significant difference. We were a bit surprised because at first we hypothesized that maybe the female will have a more carnivorous diet than the male because they have to produce the eggs. So the difference by maturity, we determined the diet for juvenile and adult, and both of them consume a lot of plants, a lot of invertebrates and a few vertebrates with no significant difference at all between the two diets. This was very surprising because in the literature you find a lot of time that the juvenile are more carnivorous than the adults, and they tend to become more herbivorous as they grow. But this was not the case in our study. The difference by period, we split our data set in two subsets, one for the reproduction period, the months of April to June, and one for the rest of the activity period, July to April. For the both periods, the consumption of plant was really high, the same for the invertebrates, but we got a significant difference in the consumption of vertebrates with more vertebrates consumed during the reproduction period. So this finding raised the question, is because the vertebrate prey are more available at this time, or is it because the species need like more energy to reproduce, to fight for female, to lay eggs, and so on? So the number of species consumed, we found 1 to 24 different species for each fecal sample. There is no significant difference between the number of species consumed between male and female, but a huge difference in the number of species between April and June, the reproduction period, and the rest of the activity period. We can look more in detail for each month with a huge amount of species consumed in June. Again, the same question as before, is it because the prey are more available or because the turtle needs more energy? Then to finally answer my main question, is the European bond a threat for other endangered species? So here you get the different species that are threatened in Switzerland, their statutes, and the number of time we find them in the sample and the percentage of it. So if you look at the vertebrates in orange, we find only a few vertebrates and 3 amphibian species, Bufo, Rana temporaria, and Pelophylax. But if you look at the number of time we found them is very, very low. And for Bufo, it was mainly at the beginning of the year in April. So we can hypothesize they feed on eggs or on tap food. But we don't know exactly. The only one we are sure it was an adult is Rana temporaria because we found in the feces some bones. Then if you look at the plant consumption, the Nymphea alba were consumed a lot, more than half of the sample contains Nymphea alba DNA. It was really easy to determine that because a lot of seeds were found in the sample. So maybe we can say, I won't say this, the pond turtle is a treat for Nymphea alba because I think it's more like a win-win relationships because the pond turtle will feed mainly on the fruit, as you see with the seed in the feces. And the study in Spain demonstrates that the pond turtle disseminate the seeds through the pond and that the seeds germinate better when they go through the digestive system. And finally, if you look at the purple species, they are the invertebrates. The number is really, really low. So we can, here is also a positive control. We find sometimes a hard part of the insect that could not be digested. So it's a positive control for the sequencing. So we can conclude that more than 85% of the species are not listed on the Swiss red list, so they are not threatened. And we can say that the impact on marginal, the impact is marginal on other endangered species, and that the feeding behavior of the pond turtle is really opportunistic and omnivorous. So the take-home message for this study, we demonstrate that the species eat a lot of plants. This was really important because on the literature, I found sometimes that the plant found in the feces were like eaten by accident. So we demonstrate that this is not the case because almost all the sample contain plants. The reproduction period was more diverse in diet and with a higher number of vertebrates. The feeding behavior is opportunistic and the number of endangered species found in diet is really limited. So the impact is marginal on the pond turtle, which is good for the reintroduction program. Like this, we can put turtle back in every pond of Switzerland. Thank you for your attention. I just wanted to acknowledge all the participants on my PhD. Without them, I couldn't undertake this work. Thank you for your attention, and I'm waiting for your question. Please be gentle. I just present my PhD today, so my brain is a little bit dead. So I see there are some questions. Are you reintroducing emis into historical habitat or introducing into new habitat, suitable habitat? So we try as much as we can to reintroduce the pond turtle in historical habitat, but in Switzerland, in the region where you can find the pond turtle, 90% of the marshland have been destroyed for agriculture, for urbanization, and so on. So when it's not possible to find historical habitat, we do renaturation work to build up new suitable sites. The second question, did you have any issue getting turtle to defect, or did most defected during your 24 hours holding period? So the pond turtle is very nice, and it's a very good studied animal because almost all the turtle defect in 40 hours and sometimes less. As soon as you put them in the individual containers, a lot of them defected in the 5 following minutes. But I would say like 10% of them did not, but it doesn't matter because we had a lot of sample and we put them back in the water even if they did not give us a sample to study. Do turtles avoid invasive and stick to food they are used to, or do they eat more of invasive as they are more abundant? Unfortunately, we did not find invasive species in the diet, except one Pelophylax, but they are not targeting invasive species. I think in the pond where we studied the turtle, they feed mainly on native species, but pond turtle is only found in natural reserve. So these habitats are really preserved and they do not have a lot of invasive species. Did you capture and compare any red slider diet? Good question, thank you. This is my next step because I got a job starting next week. And the aim is to take care of all the invasive sorry, I'm reading as I'm talking, is to take care of all the invasive turtle species in Switzerland. And one of the goals is to study the diet and determine if there is difference or if there is difference between the Trachemis and the Emis orbicularis to know if there is a concurrence in the diet. So maybe next year or in 2 years, I will present this work to the TSA meeting. So thank you for your attention, and I hope you enjoyed the talk. See you soon.
All right, well, hello everyone. Thank you for attending my presentation today. My name is Amy Bogelin, and I'm here to present to you some work that I did while at the University of Texas, Rio Grande Valley, which was comparing novel and traditional sampling methodologies to analyze the population status of the Rio Grande cooter. The scientific name for that species is Pseudemis gorzugi. So first, for a brief overview, for those of you who are not familiar with this species, it is a freshwater aquatic turtle found throughout southwestern Texas, southeastern New Mexico, and northeastern Mexico, along the Rio Grande, Pecos, and Devil River systems. You can see in that lower picture there's a historical range map, with that red being its projected historical distribution, and the yellow being actual locality information. It's very colorful. It has an elongate oval carapace with black, yellow, and green concentric circles, and it only recently received species designation in Ernst 1990, breaking from its previous classification as a subspecies of Pseudemis consina. There are a lot of conservation concerns regarding this species. To date, there's been very little research conducted on it, and it is very elusive, so it can be hard to study. However, the research that has been conducted has documented few populations of gorzugi, and a particular lack of juveniles in Texas. So this has led to a lot of concerns about recruitment for this species. They do face numerous threats, such as habitat modification and destruction. These are very heavily utilized river systems. There's been several large dams erected, as that one shown in that picture above. Also, a lot of issues with pollution, which you can see in that lower photo. And there's been some concerns about collection to the pet trade. And they do have a very small range, so they are very susceptible to these habitat changes. As a result, they maintain several current listings. They're considered near threatened by the IUCN, threatened in Mexico and in New Mexico, and they're a species of greatest conservation need in Texas. Additionally, they're undergoing a species status assessment by the U.S. Fish and Wildlife Service to determine if they should be federally listed, with a decision to be made by the end of the fiscal year of 2021. So the goals of the study were to compare the effectiveness of novel and traditional sampling methodologies while locating and quantifying gorzugi populations. In order to do this, we conducted drone, visual, and trapping surveys at 44 unique localities throughout southwestern Texas. Additionally, we developed and implemented an environmental DNA protocol at these and additional sites to determine the current gorzugi distribution, and we characterized gorzugi habitat using multispectral imagery. However, for the time constraints of this presentation today, I'm only going to be presenting on that first objective. For our drone surveys, we used a DJI 600 Pro unmanned aerial vehicle. We attached a gimbal to stabilize the camera during flight, and flights were conducted with the Maps Made Easy app and the DJI GS Pro app. These flights occurred at 30 meters above ground level with an 82% overlap and a 2.2 meter per second maximum speed over about a 1.25 hectare area. For our digital camera, we used a Sony ILCE Alpha 6000. We attached a prime lens to increase the zoom and a polarizer to cut back on glare from the water. This digital camera was manually focused to 29 meters, the ISO set at 320, the f-stop at 6.3, and the shutter speed at 1/1000. Additionally, we attached a GeoSnap Express to our drone and our camera, and this triggered our camera to take photos on a 1-second interval, and it geotagged our photographs, which were useful for analysis. For this analysis procedure, all the pictures were individually sorted through, and any photo containing a turtle was pulled aside, and the turtles present were identified and quantified. And then the GPS stamps associated with all these photos were uploaded onto Google Maps to ensure that we weren't double counting any turtles. Since we did have a very high level percent overlap with those transects, we wanted to make sure that we weren't making any erroneous counts. So we have some photos illustrating that procedure there to the right. A and B show our sympatric turtle species. In A on the left, you have the Trachemis scripta elegans and Pseudemis gorzugi on the right. And in B, there's Apollon spinifera on the left, and then the Pseudemis gorzugi on the right. And then that lower photo shows you a snap from one of our test sites of that Google Map image. So each yellow dot represents an individual photo, and each circle represents an individual turtle. So in this particular instance, anywhere between 1 to 4 photos represented one unique turtle. And those are color-coded, with white being the spinifera, that yellow-orange is the gorzugi, and that red is the scripta. For our traditional sampling methodologies, we used visual and trapping surveys. Our visual surveys were conducted with Eagle Optics Ranger binoculars. These were 15 minutes long, and we varied the order between these and our drone surveys to make sure that neither of these surveys were having a negative impact on turtle behavior. We wanted to make sure that we weren't reducing the potential counts in that following survey. All of the turtles were quantified and identified, and the observer made sure they were at least 3 meters from the shoreline at all times to reduce the impact they were having on these turtles. For our trapping surveys, we deployed 3 hoop net traps per site. These were set 1 to 5 meters from shore and baited with canned sardines. Traps were checked after 24 hours and set for 48 hours total, with trapped turtles identified, quantified, and processed. In order to compare the effectiveness of our sampling methodologies, we examined the average number of turtles detected, identified, and identification percentages for these methods. First, we had to determine our ideal sampling effort, as these different methods had different sampling units. So we constituted equal sampling events, being a 15-minute drone survey, a 15-minute visual survey, and the 48-hour trapping period, all occurring over that same 1.25 hectare area. Any effort deviations, more than 2 standard deviations from the mean, were excluded, and we additionally excluded sites and site visits with no turtle detections. We averaged abundance and identification percentages per site to avoid pseudo-replication in our data set. And as our data was non-normally distributed, we used Kruskal-Wallis and Wilcoxon multiple comparison tests for analyses. For our drone surveys, we conducted 73 surveys at 42 unique localities. This resulted in 84,441 photographs that were analyzed and led to the detection of 640 turtles, including 307 Gorzugi, 93 scripta, 89 spinifera, and 151 that were not identifiable. Gorzugi were found at 18 sites via this method. Our identification percentage was 82.3%, and our site with the highest number of Gorzugi detections had 56 Gorzugi. There's our range map illustrating our results from our drone surveys. So the way that this is color-coded is those purple circles represent sites where Gorzugi were detected, and orange is localities where they were not detected via this particular method. For a drone, we were very pleased to see it had minimal disturbance to wildlife in general, in particular with our turtles. On a few occasions, we would have turtles slip into the water. That was aerial basking, but for the most part, the drone would pass overhead and the turtles had no noticeable changes in their behavior. We did have a very unique aerial viewpoint with the drone surveys as well. There was numerous occasions where we were able to document turtles in this drone imagery that we were unable to see from shore. And this led to a very high number of overall detections. For one survey event, our drone survey detected 80 turtles, where our visual only detected 10 and our trapping only detected 6 turtles. In another survey event, our drone detected 66 turtles, where our visual only detected 18 and our trap only detected 1 all in the same survey area. We did have a high identification percentage, so we were able to identify the majority of the turtles that we were documenting in this drone imagery to species. And we had numerous occasions of behavioral documentation as well, such as mass basking, which is illustrated in photo A, subaerial basking seen in photo B, courting behaviors in photo C, and some foraging depicted in photo D. For a visual survey, we conducted 84 surveys at 44 unique localities. This led to the detection of 315 turtles, including 91 Gorzugi, 28 scripta, 25 spinifera, and 171 that were not identifiable. Gorzugi were found at 15 sites via this method. Our identification percentage was 50.8%, and our site with the highest number of Gorzugi detections had 7.7. We did have a pretty low identification percentage with this method, much lower than what's seen in a lot of prior visual surveys on different species. This could be due to the lack of animals that we had out aerial basking. Most of the turtles that we saw were swimming in the water. They're often obscured in aquatic vegetation or obscured by glare coming off that water from the sun. So it was particularly difficult to differentiate between Gorzugi and scripta on a lot of our occasions. And this here is our visual map. And once again, that's set the same way with those purple circles representing areas where Gorzugi were detected via our visual surveys. For our trapping surveys, we conducted 66 surveys at 39 unique localities. This led to 9,275 trap hours and the detection of 219 turtles, including 67 Gorzugi, 99 scripta, 53 spinifera. Gorzugi were detected at 18 sites via this method. Our identification percentage was 100%, and our highest number of Gorzugi detections was 7 for a site. Our trapping resulted in pretty mixed success for Gorzugi. We did have some occasions where we were very successful. We also had many times that we We're really struggling to trap any at all, particularly at some very high density sites. This could be due to issues with bait type. Adults are primarily herbivorous. We use canned sardines, which has been used in previous literature very successfully with the New Mexico populations, but more of a mixed success amongst those Texas populations. So they could respond more positively to a different type of bait. Additionally, we noticed a seasonal trend. The majority of our trapping occurred in these hot summer months, where in the spring and the fall, we had a little bit of higher numbers. So if we had extended our sampling season, either of those directions, we may have seen a little bit more success with that as well. And here is our trapping map, once again, set up the same with that purple representing localities where Gorzuki were detected. So for a methodology comparison, there was no significant difference found in turtle detections or Gorzuki detections, but there was a significant difference found in turtle identifications. So that top chart illustrates that there. Our drone was able to identify more turtles than our visual surveys, but there was no difference seen between drone and trapping surveys. And that lower graph shows the significant difference in identification percentages. Our trapping had a higher identification percentage than both our drone and our visual surveys. And once again, our drone outperformed our visual surveys. For our turtle detections, while we did fail to result in a significant difference, our drone did have higher mean values. This could be due to those non-parametric analyses we used, which obscure the difference in these values. But when you look at the raw data, the maximum amount of turtles detected for each survey, our drone survey that detected the most turtles detected 80 turtles in total, where visual detected 28 and our trapping detected 18. Additionally, our methods performed pretty similarly in low density sites. When there weren't a lot of turtles to be documented, there just wasn't one method clearly outperforming the others. However, for identifications, our drone proved to be superior to the visual, and our drone was the same as trapping in mean identifications, which in this instance is actually an argument for drone surveys, as trapping is always going to result in perfect identification. But essentially, the drone proved that it could conduct these species-specific surveys, which really is essential for wildlife management. You're not going to be as interested in how many turtles are in a particular area, but how many of each particular species, especially when you are looking for these species of conservation concern. So for our conclusions, our drone was minimally invasive and it had a unique aerial viewpoint, which led it to the opportunity to detect a lot of these turtles that these other methods may have been looking over. However, there are numerous requirements with this particular method between different licensing and permits that are specifically required for the drone. And it is a newer technology, so there are some technological issues that are still being worked out. However, there has been great progress made in this particular area, even just from the start of our study. And it has continued to become more feasible to implement this new technology as it becomes more popular and just more widespread. For our visual surveys, they were low effort, low cost, and quick and easy, which is why it's often one of your first line steps that you're using when you're accessing an area. However, they are subject to observer bias. Your results are only going to be as good as that observer is at locating and identifying turtles. And in our instance, we did have a pretty low identification percentage. We weren't really able to get that species-specific information we're looking for. For trapping, one of the main benefits of trapping is you have a turtle in hand, so you're always going to be able to identify it. You have the opportunity to collect population health data, tissue samples, vouchers, certainly a lot of unique benefits with this particular method. However, it is labor intensive and it can become expensive because of that. It's subject to numerous sampling biases. There's all sorts of literature out there about how different turtle species respond to different types of bait age, whether or not there's a turtle already present in a trap. Some species just really don't trap well in general. And trapping is also unfortunately subject to trap theft. So depending upon what your particular needs are, what particular data you're trying to obtain, and what your resources are, is going to depend on what your method, I guess, would be most suitable for your use. However, we found that that drone did have a lot of unique advantages and certainly should be a top contender when you're looking for a way to survey for freshwater turtle species. But being able to combine all these different methods together, we were able to have our most thorough assessment of this area. And we created an updated range map for this particular species. You can see there to the right, just a few more purple circles than you had seen in any of our previous individual maps. But being able to combine all this data certainly was advantageous in being able to survey for this species. And with that, I'd like to acknowledge the University of Texas Rio Grande Valley for the opportunity to partake in this project, as well as the Texas Comptroller Office for providing funding, and several outside organizations and individuals who all helped assist in the project and permitted sampling on their different properties. And if I have any questions, I can answer those, and I can also always be reached for follow-up via that email address below.
Do you see your questions in the chat?
Yes, so just pulling those up right now. All right, so question 1. How far below the water surface could these turtles be detected? Wondering if this could work for estuarine sea turtle surveys. So I don't have a particular measure on that. A lot of our surveys occurred in the Rio Grande, the Pecos River, and those, I don't have the exact depths of those, but there were several occurrences where we were able to actually document turtles that were all the way on the substrate of these river surfaces. It's certainly going to be dependent upon how turbid the water is. I mean, from one sampling event to the other, we have certainly had differences in the amount of turbidity. But if you did have clear water, you should certainly be able to see several meters below that water surface. A lot of sea turtle work has actually already been done with drone surveys. Sea turtles are much larger, at least most of them are, than a lot of these freshwater turtle species. So they do have that going for them too. But I know it has been used already to document a lot of sea turtle surveys and has been pretty successful with that. Question 2, you mentioned possible seasonal effects on trapping. Did you detect any seasonal difference in your other 2 methods? Yes, I'd say we had some of that as well, particularly noticeable, I guess, in both our drone and our visual surveys, is in those earliest sampling dates and in those latest sampling dates, we did have a few more turtles aerial basking, where in our bulk summer months, it was a very infrequent occurrence to actually have that happen. That picture I showed of that mass basking, there was 26 Gorzugia on that rock. That was very early in the year. When we came back to that site a few times later on, at the same time, we didn't witness that same thing, which we figured was just due to the much higher temperatures that you're experiencing in South Texas during those summer months. Question 3, do they plan on using this method to study any other species anytime soon, more cryptic and secretive species like the soft shells and muds, etc.? I do not know of any plans to study more cryptic species with this method. Certainly, your more success is going to be for these large basking turtles, just because smaller you have, the more difficult it is going to be to detect them via this imagery. And if anything you have that's like burrowing in mud, you know, it's just not detectable on the surface as something flying overhead, you're not going to be able to see it. One thing that I have heard of, but I don't think it's actually been used to date, but for some of these more cryptic species would be potentially using thermal cameras and actually looking for those thermal heat signatures in areas where you might not be able to visually detect them. So that would potentially be an opportunity as well. But we did document our soft shells in our drone surveys, and several occasions they were often harder to see just to their habitat use, but it is certainly possible. In question 4, isn't it difficult to identify turtle species only with carapace and top of heads in the pictures? What are the key features used to identify each species? So yeah, certainly the more different in overall appearance your turtles are, the easier it is going to be to differentiate them. We were lucky in that we only had 3 sympatric species in these images, and the Gorzugi had these very bright, unique yellow bands on the top of their head, which was one of our top things that we were looking for. The Spiny soft shell obviously is very different in appearance also ever between the colorings, just that elongated snout, the red-eared slider, and many occasions we were able to actually see that red marking by their ear. Another thing with Gorzugi is they have bright orange webbing between their, I guess between the, like on their feet. So we basically tried to use a combination of these. If there were any instances where we weren't 100% sure on what the identity was, we did classify it in that unidentifiable. But for the most part, like I said, we generally had identification percentages in the 80%. In question 5, were you questioned by Border Patrol officers while flying drones over the Rio Grande? Yes, we frequently saw Border Patrol. I would always call them beforehand to alert them of our presence, but that seemed to never actually make it out to the officers in the field. However, the Border Patrol was generally very happy to see us, just doing, you know, turtle research. Many of them actually showed us pictures they had of the species we were looking for at our site, and they were always very excited to see the drone fly. So we certainly didn't mind having them around. And I believe that is all my questions. So thank you all for attending, and I'm going to pass it off to the next presenter.
Hello?
Hello, Camilla.
Can you see my video?
Your share screen is not up yet, but we can see you.
Oh.
There it is.
Okay. Can I start?
Absolutely.
Nice. Hello, everyone. Thank you to come here today. And let's talk about the acknowledgement of behavior patterns related to the mass nesting and mass hatch of Podocnemis spansa to develop conservation strategies. The speed human population increase in Brazil and has been threatening the Amazon ecosystem and their wildlife. One of the main threats is the growing and the uncontrolled demand for bush meat. And unfortunately, turtle is not an exception in this scenario. Actually, giant South America river turtle, Podocnemis spansa, is classified as critically endangered by the UCN group specialists, mainly because the illegal consumption and the traffic. In the Amazon, every year during the dry period, turtles become to be more vulnerable, especially for the illegal consumption and the traffic, because they stay in groups and move in groups. They move from the feeding areas toward the riverman waterbeds, where are the nesting sandbanks. To try to reduce the pressure in Podocnemis spansa, with the support of the Fundação Boticário and ICMBio, in an important protected area located in a tributary of the Amazon River, we aim to improve methods of protection and management of nesting areas using aerial images obtained by drone, acoustic communication, and environmental data to build statistical models that we can predict the mass nesting and mass hatching period in the reserve. We started this project last year during the nesting activity. And when we went to the field, we didn't have much information about how to use drones. We read few papers about sea turtles, but we didn't have much information with freshwater turtles. And then the first thing that we decided to do, it was to do some tests to know the proper altitude that we can use the drone without annoying the turtles. Also, we classify the turtles in 3 different behavioral categories. No movements, brief movements with the head or body, and sudden movements. When they are on the nest on the beach, they go back to the river, or when they are in the river, they just dive. We started in 150 meters, and we didn't observe any disturbance from the animals. And then we come down with the drone until 50 and 25 meters. And also we didn't observe any disturbance of the animals. But in 15 meters, we observed a brief movement with the head, and in 10 meters, they dived. But when they are doing the nesting activity, it was possible to use the drone and fly with the drone until 10 and 5 meters of the altitude. We just observed brief movements with the head. Also, during the nesting activity, we had the opportunity to observe a little bit of the female's dynamic in front of the nesting beach and on the sand on the nest beach. Here, we have a sequence of 5 days photos. We took all those photos at the same days. On the first day, if you pay attention, we have a lot of females in front of the beach. It's around 42, and few females on the nesting. But at the following days, the number of females in front of the beach and on the beach start to reduce. From those photos, is it possible also to observe that the females, they use exactly the same area on the nesting to nest every day. But there is, they use the same area during the nesting activity. But each day we can observe that there is a variation in the location of the nest. On the day, between the day 3 and 4, is it very easy to see the difference. They just use each day part of the nesting area. Also, we collected some data during the pre-hatchling period. In this period, we performed 12 those flights in a 1,500 transect parallel to the nesting beat in 4 different times, 7, 10, 1, and 4. And during those flights, we try to obtain the best images to study population estimates and do the behavior analysis. In each drone fly, we collect the images in 4 different ways to allow us to cover all the study area. Here, I have just a quickly video to show you, just for you see how is the drone performance. Drone is a good tool to population estimate, to understand, to do behavior analysis. We can do zoom in, zoom out, stop the images. Here we have a large group of turtle together. And all the fly we made in 25 meters of the altitude. And then the smallest number together of turtle we found at 7 o'clock in the afternoon. The largest number of turtle together we found at 1 o'clock in the afternoon, 9 to 1 turtle together. But we didn't find any difference between the methods that we used to get the images for population estimates and behavioral analysis. Also, during the pre-hatchling period, we could observe in 3 different occasions courtship behavior. But unfortunately, it's very hard for us to identify the sex of the animals. At this period also, we are recording the females, we are recording the female sound under the water, and we made the recordings at the same time than the drone's performance. Here is the equipment that we are using. This is the recorder and the hydrophone. This is the microphone that I will talk a little bit later. And here is a quickly video just to show the drone performance when we are recording the turtles under the water also. We are recording the hatchlings and embryos inside the nest. And until now, we just find the 2 types of sounds. 2 types for the adult females and just 1 type for the hatchlings. I can play, but I'm not sure if is it possible to hear the sound. I hope people could hear the sound. And then we are still collecting the environmental data, water temperature, nest temperature, and level of the river. Next week, we will go back to the field again to still getting the data to build our statistical models to predict the mass hatchling and the hatchling period. But until now, we already know that drone can fly above 25 meters without annoying the animals inside the water, and 5 meters when turtles are nesting. Drone is a good tool to study turtle behavior and population estimate. And Podocnemis spansa also use sound to communicate in other type of water, in this case, what water. And before I finish, I'd like to thank you to Fundação Boticário and WCS, and thank you. Questions?
Sit tight for just a minute and we'll see if we have any coming in. All right, Camilla, are these sounds new types of sounds inside of the Expensa repertoire?
The sound types are the same, but the frequencies are different.
Okay, gonna hang on for just another minute to see if we have another question coming in from YouTube. Are the drones that you're using very expensive?
Expensive? It's a little bit because we bought the drone in Brazil. It was expensive, but I think is it possible to use drones with a cheaper price than our drone?
Cool, cool. Another question, do you have recorded sound?
Yes, yes, we have been recording the sound.
Another one is, are these turtles legally protected against poaching?
Yes, they are protected, but not every time the laws work in Brazil. They live in a protected area.
And that was the last one. Thank you, Camilla. Up
Thank
next,
you.
Up next, we've got Michael Arendt. You ready to go, Michael?
Just about. Can you see my video? I thought you wanted me on video as well. Something happened here with the screen. All right, I think we're on there. And is my presentation up?
Not
Hello?
yet.
Not yet.
Yeah,
Okay.
but I'm sure that it will be again.
Thought I hit share screen.
There you go.
Share it.
Here it comes.
All right. How about now? Looking solid?
Perfect.
All right, great. Well, thank you very much. On behalf of my co-authors, we're very excited to present an exciting breakthrough in the design of bycatch reduction devices, or BIRDs as they're more formally referred to. And these are used in wire mesh crab traps, and drowning in crab traps is a top threat for recovering diamondback terrapins throughout their distribution range, which is Texas to the northeast United States. And throughout this range, they are listed as species of greatest conservation need by the Wildlife Action Plans. The bird that I'm going to talk about today was, and it's the red one in this photograph here, it actually very closely resembles the original bird first proposed by Roger Wood in 1997. And that was about a decade and a half after the drowning of terrapins was first reported in the literature. And since that time, numerous studies have been conducted, and what the birds have sort of evolved into are these orange rectangular shapes. These are actually required now in some mid-Atlantic states, but in many respects, that's unfortunate, because when you look at it from a conservation perspective, when terrapins enter a crab trap, they do so head first, and as such, they're almost twice as wide as they are tall, which means that only the largest terrapins are excluded, and there's a lot of wasted space with this device, which means that crabs can just as easily escape as they can pass through. The reason these devices were so wide is to try to appease crabbers, because Roger Wood's device didn't catch quite as many crabs as he had hoped. And folks were thinking, you know, in terms of the carapace width of the crab, maybe the device needed to be larger. It turns out when you look at how crabs enter traps, they do so essentially sideways, just like you see them swimming in the water. And so it's their carapace length that it's actually that horizontal dimension. So taking this into consideration, we were able to design a bird that was not only considerably taller, especially with these rounded top and bottom edges to accommodate that body shape, but substantially narrower. And this allows us to improve the conservation benefit of the excluder device. So the work that I'm going to talk about today is published in the North American Journal of Fisheries Management from 2018. So I'm not going to go into a lot of detail other than to say that the analysis approach that we used was a 5-point scoring system where we assessed the probability of entry for individual crabs, 736 crabs, or the probability of exclusion for 985 individual diamondback terrapins. And we assessed the probability of these individuals to pass through almost 8,200 different theoretical bird sizes. These bird sizes represent the 1 millimeter combinations between the smallest and the largest width and height among all the bycatch reduction devices that are reported in the literature. And when you synthesize this data for each of those bird types, you see shown here the probability of entry for crabs or exclusion by terrapins on the X, and the percentile distribution across those 8,200 bird sizes. And what you see here for terrapins in the top series is a relatively smooth continuum with regards to will this bird exclude terrapins. And fortunately, with respect to crabs, you see that most of them are at 100% entry. So very few of these theoretical designs would have substantially reduced crab catch. So that's a positive for encouraging the use of bycatch reduction devices. When we look a little more closely with regards to predicted observations versus actual catch in these birds, where we standardize each bird tested in a study relative to the number of animals caught in the control and what proportion of the control crabs the respective bird should have allowed in the bird traps, we see that most of these symbols fall along the dashed diagonal line, which is great news. That's telling us that the birds basically are performing as predicted for crab catch. When we take a little closer look at the terrapin scenario, what we actually find is that a lot fewer terrapins were caught in traps with birds than would have been predicted based on the size of the birds. And this has led a lot of it's almost a false positive because it's led to a lot of sense that, you know, this is the right size bird, when actually it probably was really more of a behavioral deterrent, as suggested by direct observation studies conducted both in Virginia and South Carolina. So, you know, with that in mind, going forward, we want to make sure that we have the best mechanically sized bird so that we're not relying on behavioral deterrent alone to keep animals that we don't want to drown out of traps. And what we found is that aperture area is actually the best indicator for terrapin exclusion. And I've shown bird area on the x-axis and the probability of exclusion on the y. And the dashed lines are a general continuum with respect to bird area. And the symbols denote the mean and 95% confidence intervals for both our bird design as well as the 3 most widely tested bird designs in the literature. And what you see here is without a doubt, we have superior terrapin exclusion probability because of the sizing of this bird. Unfortunately, we're one of the few designs where you actually see a reduction in crab catch because our bird is so narrow. And this reduction in crab catch was also reported from a companion study in Virginia. And so we took this into consideration. We've since made our bird just 5 millimeters wider. And as a result, our crab catch goes up to comparable to those orange birds. And we only see a 4% reduction in terrapin exclusion probability. So again, we're still dealing with the best bird on the market, if you will. So the next few slides, I want to talk about some success stories where other folks have taken our new and improved excluder and really found some further encouraging results. The most exciting news comes out in New Jersey, where last summer, John Wynick and colleagues tested our bird relative to those other orange birds, and as well as relative to traps with no birds. And what they found is that almost half of all the legal sized crabs that they captured were in the traps outfitted with our bird, and the remainder were spread across those other 3 trap treatments. John told me that they plan to move forward with not only publishing these data, but also pushing for improved terrapin conservation efforts in New Jersey, which is fantastic because New Jersey definitely deserves the title of birthplace of terrapin conservation. In North Carolina, immediately after our paper was published, I was contacted by their management folks, and they said, we'd really like to move forward with your bird. And I said, hey, if I learned anything from our analysis, it's that I should have thought about morphometrics first and gone out and physically tested it second. And so we partnered with Amanda Southwood Willard at UNC Wilmington, and she was going to try to work with commercial crabbers to test it and get measurement data and whatnot. But the combination of tropical systems moving through the epicenter of East Coast hurricane landings, as well as just the general apprehension when they saw the small size of this excluder. So that crab measurement aspect really hasn't happened yet, but Amanda and her team were able to move forward with getting terrapin measurements, which showed that our bird would be effective in North Carolina. And just this spring, I received word from the North Carolina Marine Fisheries Commission that they are moving forward. They have adopted a management strategy to use birds in crab traps. This is not a statewide decision, but there will be smaller specific terrapin management areas that will be implemented starting in 2021. And this really was based on a combination of our bird paper, our bird design, as well as the UNC Wilmington terrapin morphometrics. So really encouraging news, especially when you consider how long this has been tried. People have tried to push this forward in North Carolina, and as well as the significance of the North Carolina blue crab fishery relative to total East Coast landings. To the south of us in Georgia, we've partnered both with fishery independent researchers as well as commercial crabbers, all of whom reached out to us and said, we've heard about your bird, we'd really like to test this thing. Unfortunately, I don't have a lot of details from those efforts to share, other than that the preliminary results from the laboratory portion of the Georgia study do indicate that higher crab retention with traps outfitted with the bird. So again, reinforcing that physical observation from New Jersey. So with all of these exciting developments happening up and down the coast, you might be wondering when South Carolina is going to be the next state to implement terrapin conservation measures. All I can tell you is that decision occurs well above my pay grade, but we are continuing to do what we can to support that effort and instill confidence in our leadership and the folks that ultimately make those decisions. Where we are focused right now is determining the extent to which crab and terrapin morphologies may differ across South Carolina estuaries. Remember, just the difference of a millimeter or two has a huge impact on the predicted entry of a crab into the trap with our bird. And we know from partnering with volunteer, recreational, and commercial crabbers throughout the state, as well as a pilot study where we went to different crab houses and actually measured crabs, that there does appear to be a slight difference where crabs to the south of us are, in the southern part of South Carolina, are just a little bit wider relative to their height. And that is reducing their probability of entry. So we need to figure out, what is really the best either universal size, or maybe we have different bird sizes within the state. So we had actually pitched a proposal to generate that type of information through the State Wildlife Grant Program. Unfortunately, due to budget cuts, it was not recommended for funding in the coming year, but we do plan to continue to piggyback on other projects to try to generate that data set just a little more slowly than if we'd had directed funding. And of course, if we determine that the variability is extensive, we want to look further into why that's the case. Is it a genetic thing? Is it a salinity gradient thing? So on and so forth. So in closing, I'd like to put a different spin on the bird. We're gathered here today as terrapin and turtle aficionados, but if we really want to see widespread bird use, we need to get out of that comfort zone and embrace promoting the bird as a way to improve the vitality of the crab trap fishery itself. Certainly, we know from the New Jersey work and to a less extent in the Georgia lab study that when properly installed in the funnel, our bird is associated with retaining more legal-sized crabs. That is money in the pockets of the people fishing those traps. That's an incredibly positive message to put forth if you want to increase compliance. Secondly, we know that more than 40 species are incidentally captured in crab traps, but terrapins, because they're air breathers, and perhaps because they're charismatic species, tend to get a lot of the attention. And so if you're a crabber that's out there and you don't see a lot of terrapins, you kind of begin to wonder why this is such a big deal. And so we really need to embrace the bee bycatch in bird. The other thing is that we know the absolute largest crabs that are out there to be caught will not fit through our bird. And these are unfortunately the most economically valuable crabs to the fishers. And so we need to figure out a way to promote and reward crabbers, promote it as a sustainable fishery and reward crabbers who are minimizing the bycatch of all these different species, especially since we know traps can be lost for a variety of reasons. And so we need to come up with an economic model that really supports compensation for using the device to offset any economic loss from not catching the biggest crabs that are out there. And then another angle that just occurred to me this year after a recreational crabber in Texas reached out to say, hey, I've got river otters that are raiding my traps. Do you think your bird would help keep these river otters out of my traps? Is that maybe it's a way to reduce predation and poaching by other species that are often protected, like the river otters or dolphins or sea turtles. Now, ultimately, the crabber elected not to use our bird because he regularly catches crabs that are 8 inches and larger. He didn't want to lose those, even though he's a recreational crabber, not a commercial crabber. But he was kind enough to send along this very powerful image of an 8-inch point-to-point Texas Jimmy blue crab passing through our excluder device, which I think is an amazing testament to what you can achieve when you take the morphology of your target species into mind and then design your excluder around that. I know we're short on time, so I'll wrap it up here. I'd like to thank the U.S. Fish and Wildlife Service State Wildlife Grant for funding various aspects of our research that allowed us to ultimately design this device through 2016. Lots of great folks that helped us secure funding and manage the grant. In addition to my co-authors, lots of volunteers helped us out there in the field over the years, measuring individual animals. And with that, I will be happy to take any questions. And I'm going to try to pop up my chat box here so that I can attempt to answer. It's a little technologically impaired here right now.
I can give you your questions if you like, Mike.
Yeah, I can't get back to the wide screen here. I'm not sure what's going on.
No problem.
Go
You have
ahead.
2 questions at the moment. Question number 1. Since female terrapins are much larger than males, could birds work only to exclude females and not males? And if so, does this skew the sex ratio of the population?
So the answer is that adult female terrapins are much larger than males, but they don't start off as much larger, and they have to pass through annual survival gauntlets that every year they would fail if the only bird out there was the one that only protects the adult female. So it's really important to protect all of these life history stages leading up to that maturity stage. And if you're dealing with an adult only population, then yes, you would be excluding the males at the sake of conserving the females. But again, they don't start off that size; they have to reach that size, and that's why having the properly sized bird from the onset is the best thing to do for conserving this species.
All right. The second question, I may be off on this, but would this work for sea turtle hatchlings as well, or older young sea turtles?
I don't know. I would say no, because again, a sea turtle hatchling is not much different than a diamondback terrapin. Certainly a juvenile diamondback terrapin. So we know that, again, I had 62% exclusion down to about 58% exclusion when we made the device wider. And so that means that, slightly less than half of terrapins that are out there, based on the overall size distribution in the area that we conducted our study, would pass through this excluder. And so most sea turtle hatchlings would be small enough to go through here. I'm also not aware of instances where sea turtle hatchlings are entering crab traps, but if somebody has information on that, I'd love to hear about it.
Good deal. That's the last question, Mike. And
okay, I'm going to stop my share and
thank you very much, Mike. Up next is Ellery Ruther Lassiter.
How is that looking?
Perfect.
Awesome. Okay, well, thank you for having me. Again, my name is Ellery Ruther Lassiter, and I'm a graduate student at the University of Arkansas. I'll set my timer here. And today I'm going to be talking about movement and space use of spotted turtles. There's spatial heterogeneity in landscapes, and differences in landscape suitability can result in differences in resource availability, which can in turn influence the movement patterns of organisms. So when movement between patches occurs, it can vary across time, frequency, and space, based on the intrinsic values of the individual, such as sex, age, or size, and extrinsic values of the landscape, such as resource quality or weather that it might allow for or impede movement. And this is, of course, going to be really important in dynamic environments, such as a wetland ecosystem. Wetland ecosystems tend to be both spatially and temporally variable over time, causing wildlife to have to utilize several different wetlands in a given season. Movement between wetlands is vital for maintaining population stability, and therefore characteristics such as wetland quality and connectivity are likely to influence demographic processes. The ecology of semi-aquatic freshwater turtles is especially complex because individuals often make long-distance movements between a variety of habitats for mating, foraging, basking, estivating, and overwintering purposes. And one of such species is the spotted turtle. The spotted turtle is a relatively small semi-aquatic species with a wide-ranging distribution along the East Coast from Florida to Maine and in disjunct populations along the Great Lakes region. And often overlooked, turtles are actually one of the most threatened vertebrate orders in the entire world, with nearly 60% of all species listed as threatened or endangered, which of course is the impetus of this entire conference. And this is in part due to a set of unique life history traits that many turtles share. Spotted turtles specifically are long-lived, with females reaching over 100 years old and males over 60. They've delayed sexual maturity, in which males and females don't reach reproductive age until over 10, and fairly low fecundity. Females lay typically about a clutch a year, but they may not reproduce every year. And all of these traits combined make population stability especially sensitive to adult mortality events. Even the loss of just 1 or 2 individuals can lead to population decline or even local extinction events. And unfortunately, spotted turtles are declining across their entire range. In Canada, they're listed as endangered, and in the US, they're listed as a species of greatest conservation need in every state they occur in. And they were petitioned for federal ESA listing in 2015. And that's where my project comes in, because in response to that petition, the DoD funded a project to proactively study spotted turtle population and distribution so they would be better prepared to manage and avoid conflict if the species is indeed listed under ESA. One of such projects was understanding how spotted turtles use and move across heterogeneous landscapes. And so specifically, I wanted to look at the influence of extrinsic habitat and climate characteristics and intrinsic turtle characteristics on movement and space use. And I used several different measures to understand the system a little bit more fully. To understand movement, I looked at inter-wetland movement, so when a turtle moved from one wetland to another, or the number of days a turtle spent in a given wetland, and then of course the distance a turtle moved. And to understand space use, I looked at the number of wetlands a turtle occupied in a given season, as well as its home range size. My study site is located in Virginia, where I had 3 different subpopulations, each with their own patchy wetland mosaic. Of course, these wetlands differed in size and distance from one another. And again, I was interested in looking at the movement between these different patches, as well as across the landscape, and then space use and general home range size in the system. In order to get this, I equipped adult and juvenile turtles with VHF radio transmitters and tracked them every other day during their main activity season, which in Virginia is approximately March through August. For home range estimate, I used 3 different estimates, but for the purpose of this talk, I'm just going to present my autocorrelated kernel density estimate. And then to look at the influence of different covariates on movement and space use, I fit generalized linear mixed models, with the predictor variables being intrinsic and extrinsic factors, and the response variables, of course, being those measures that I just described. And so those intrinsic and extrinsic covariates were sex, age, and size of a given individual, and then climate and habitat characteristics around the given wetlands. And in general, I expected to see differences in sex and age due to differences in life history strategies. Males locating females, females nest searching, and juveniles kind of sitting tight and reducing their movement because they generally have a higher predation risk, with of course some effect of climate across a given season. However, surprisingly, I ended up seeing a large effect of climate on movement covariate or movement estimates, while I only saw an effect of age on space use. So let's take a closer look. Seasonal effects that mirror what's in the literature. So you can see on the top left that turtles tend to move more in the more moderate part of mid-spring. Turtles were also more likely to move between smaller, more seasonal wetlands than larger, more permanent wetlands. And likewise, turtles are more likely to move between wetlands when the wetlands are more shallow. And that is likely hydrology related because the seasonal wetlands tended to be more shallow, as I just mentioned, and also climate related because as wetlands are drying up, turtles are having to move in between those different wetlands. And then lastly, turtles are more likely to move with rainfall events, and that's possibly to reduce risk of evaporative heat loss or just in anticipation of getting back to a preferred wetland. Days spent in wetland is kind of the opposite side of the same coin, so same exact estimates here, but the different effect. The turtles spent more time in wetlands when weather conditions were unfavorable. They spent more time in larger, deeper wetlands, and they spent more time in one place during periods of no rainfall. And this is particularly demonstrative of their estimation behavior. Distant moved was related to interwetland movement. So again, we see that strong seasonal effect where they're moving more mid-spring. But we do see a slightly different influence on number of days since last precipitation event, where we see turtles had to move farther when it hadn't rained in a while, presumably when wetlands are drying and they're further and further out to find a wetland of their preference. And then, of course, intuitively, turtles move farther when wetlands are farther from one another. And here we have stills of movement paths for each sex and age class across one given season with home range estimates underneath. And starting on the left, for juveniles, we can see tightly clustered movements, while adult counterparts seem to move a little bit further. For females in particular, you see the nest movement, that huge circular area before they return to their activity center, where they're most highly clustered. And then males also demonstrate an activity center, but you see that that is a slightly larger cluster with more clusters than the females. And so if we take a look at the estimates themselves, we do see that juveniles have a smaller home range and they occupy fewer wetlands than their adult counterparts. But surprisingly, we actually see no differences in males and females, either for number of occupied wetlands or for home range size. However, despite the lack of difference, I will note that this is actually a data set that does not include 2 outlier males. And so if we add in those outlier males and we take a look at the difference in home range, so moving to top right, we see that there actually is a difference between home range size specifically with males and females. And this difference is actually driven only by 2 outlier males who made 2 long distance movements, one 2.3 kilometers and one 3.4 kilometers respectively. So whether or not there is actually a difference between males and females is perhaps unknown. These turtles are capable of making some incredible movements, but who, why, and how often those movements take place is uncertain at the moment. And so while it should be obvious that climate is, of course, an important driver of movement and dynamic ecosystems, it is interesting that it is potentially the only driver of such movement, while space use is driven by age and not as influenced by climate. The connectivity of these systems is obviously very important to take into account. There was a lot of movement in between these different wetlands as they're responding to how their climate or how their habitat is changing. However, there is potential movement between, or there is potential movement between subpopulations, and so that has to be taken into account. Connectivity within and between subpopulations is going to be really important for the perpetuity of this species. So moving forward, what are the next steps? I plan to implement a genetic analysis. I've taken genetic samples across all of my subpopulations, and we're hoping to identify who are these individuals moving between the subpopulations. And specifically, are they within certain age classes? Are they within certain sex classes? And how often are they moving between the populations? And that way we have a better sense of how far these animals are moving and what kind of space use, what kind of connectivity, and what time period are these individuals most susceptible. Because again, tracking is only one snapshot of their lifetime. And so luckily using sort of newer genetic methods can help us to determine the actual level of dispersion and connectivity between populations. And then of course that these systems might be very susceptible to climate change. And so trying to look at how long these wetlands will stay full and how often they're going to pop in and out of the landscape will help understand what the susceptibility of this species is in our dynamic climate moving forward. And so with that, I would of course acknowledge the different institutions that I've been involved with and my funding sources, as well as of course people who have helped me in the field. And with that, I'll take any questions.
We'll sit tight here just for a second and see if we have any from YouTube. Do you have your, would you like me to read
I do have my chat open.
Very good, thanks. Do you see the question?
Okay, So the first question was, were there any barriers to movement at the study site you had to incorporate in home range estimates? That's a great question. I thought that different road classes versus like dirt or paved road might be a barrier to movement, or rivers might be a barrier to movement, but I saw turtles cross all of those with ease. This is especially evident when I saw the turtles move for the 2.3 and 3.4 kilometer movements. They went into a large bay, they went into different wetlands, they went into a forest, and then likewise, very anthropogenic features that I thought would stop them, but didn't. So perhaps no. Question 2, did you look at the surrounding land use and how could that have affected overland movements? I did look at land use. Specifically, I included percent development in the model, and it was not influential. The movement did take place on a large area of protected land that is multi-use, but there was not enough difference between the subpopulations to really see an effect of land use, unfortunately. Question 3, reintroduction is occurring near me. What is the recommended minimum size to reintroduce into its minimum size in acres? Sorry, this is written a little weird. That's a great question. I have not dived into that. I do plan on conducting a PVA analysis, a population viability analysis, to better understand that relationship between size and population viability. Many of my turtles had very small home ranges, but there was a lot of variation. These large movements were potentially just dispersal events and not obviously reflective of the actual home range size capability for this species. But in general, maintaining a large matrix of wetlands so that turtles have a lot of different options as the habitat changes across the season is obviously very important. The next question, in some populations of spotted turtles, they become terrestrial and dormant in the summer. Did your turtles do this, or did they always go to a wetland and stay active? I saw a little bit of both, as the weather was different every year that I tracked. I did have 4 years of data. Predominantly, they did move onto land and estivate and really didn't move very much. They might come out during a rainstorm event to an adjacent wetland and feed or move around. I wasn't always observing them. But other years where the wetlands stayed full and wet the entire year, they still nonetheless estivated, but in this time they estivated aquatically. So they do have a period of inactivity, whether that's terrestrial or aquatic, at least in Virginia. And then lastly, is there any significant human-related driver for this movement? You might say climate change is perhaps a human-related driver. Again, I saw a huge influence of climate on spotted turtle movement, and less so from landscape and less so from those intrinsic effects. So these animals are moving in response to their weather. They live in very dynamic systems, and so they have evolved movement that matches that. And so as their dynamic system continues to change, it'll be interesting to see if their behavior is plastic enough to change with it.
And that was the last question. Thank you, Ellery.
Thank you.
Now we're going to see if our next speaker is ready to go. I think she is.
Yes, hello. Excuse me.
We can hear you.
Hello, everybody. This is Gracia Gonzalez Porter, and I am going to present a classic work on Dermatemis, Maui. Let me start my presentation. I don't know if you can see it. Can you see it as, okay. And I need.
We can see
the chart. Okay. And okay, no. Wait 1 second. Okay. Yes. So, one second, just let me move this. Okay, so I'm going to talk about the home range migration habitat used by Dermatemis maui in Rio Sendales, La Cantun, Selva Lacandona in Mexico, in Chiapas. As we know, Dermatemis maui is a species that is critically endangered, and it is the last survivable species from the whole family. So if this species gets extinct, the whole branch will be extinct. So, okay. The study area is in the Biosphere Reserve of Montes Azules in Chiapas, in this area, Mexico. In 1980, a huge, large catch of 250 turtles were seen in the La Cantun River. They eventually got confiscated by the military and taken to Frontera in Chiapas. And then the people from Fauna Silvestre took it to Tuxtla Gutierrez. So you can see all the turtles in this cayuco.
Gracia, I'm sorry to interrupt, but you're still in
Yes.
presenter mode. And if you could go to display settings.
Display settings, yes. So what do you want me to do? Swap presenter?
Swap presenter view.
Okay, is that better?
Perfect, thank you.
Okay, I'm sorry. So, because I have the pictures here. Okay, so this is the study area. I mean, from the waterfall where the animals were living, and they were like moving from this area, I mean, up to the river and the waterfall in Rio Sendales. Sometimes the animals can shed the whole whole scutes if they're older enough. The scutes from the shell get fused, so sometimes you can see all these the scales shed. This is a female. As you can see, this animal has a lighter color head. So it's a female, and the plastron, you can see the smaller tail, and it is all white. So in Mexico, the name of the species is white turtle or tortuga blanca, because they don't have any patterns on the plastron. This is a male. As you can see, the male has a brighter color head. It can be orange or yellow. Actually, this is in Philadelphia Zoo when they have the animals there at field. I mean, you can see also the yellow head, and this head gets brighter with the reproductive season. The males also have a larger tail, so on the penis, is this one. In the throats, they have this papilla where they're very vascular, so that allows them to get oxygen from the water. Here we can see an animal that is passing oxygen through their nostrils and through the mouth, and in that way they get the oxygen so they can breathe under the water. So here is the bubble. This is one of the Dermatemis maui, a nest very close from the water, at the water level. Sometimes these areas get floated, so the eggs can be under the water, and they have this diap house. So they stop the development of the embryos inside of the shell. And they can restart the development after they get dry. So this is a closer look, and as you can see, the nest is wet and they can be under the water. This is an area with shallow water and very slow moving water where they live. And if you can see, this is full of vegetation. So the Dermatemis maui is a herbivorous species, so they can feed on these branches and underwater vegetation. This is the study area, the river. This is the Sendales River, and all the way to Laguna Oaxaca, where this study was taking place. Okay, so the aim of this study was to determine the home range and activity patterns of Dermatemis maui in Rio Sendales on April 1992. They were attached 10 VHF transmitters to 2 adult males and 8 adult females, Dermatemis maui, from Rio Sendales downstream from the waterfall. We saw the waterfall in another slide, migrating upstream. The turtles were monitored over 14 months. The first 4 months stay in a slow current area and lake areas, feeding on leaves, lianas, and other terrestrial plants, and overhanging the river. So we saw the vegetation. It is easy to document that the Dermatemis maui is feeding there because of the marks, the serrat bites on the leaves and the floating scat. It didn't, I don't know what is going on, but it's not moving. And second, well, okay, let me see. Okay, so 4 females stayed together in this area. 1 female moved downstream 600 meters and moved 400 meters upstream in the San Pedro River in 2 days. 5 females and 1 male stayed together, moving 50 to 100 meters up or downstream until the water was rising high in the Sendales River. By the October 26, 1992, 2 females remained in the San Pedro River, or the Rio San Pedro. The remainders of the males and females left on the Lacantun River by the end of October and never returned. 1 female left Rio Sendales and went 2 kilometers upstream in the Rio San Pedro. This is the Rio San Pedro is a warmer, shallower, and less current river. There are known nesting sites here, as well as in the shallow backwater areas where we observe hatchlings in groups of 8 to 23 foraging from the 1986 to the 1998. Okay. Dermatemis uses the Rio Sendales during the low water season, during the dry season, and the rest in the whirlpool areas in May when they're shedding the scutes, and this is when the raining season starts. After the raining season begins, and along with the rain, the nest, they live in the Sendales and they travel 7 to 12 kilometers downstream to the Lacantun and disappear 20 or more kilometers up or downstream. In March 1997, this population was declined to only 3 adults and 2 juveniles, they could be captured in 5 days. These species need larger reserves in order to protect their area, their home. So it is very important to protect this area and to make it bigger. When we returned in 2003 and 2016, we didn't find any turtles. So this is very, very important and very sad. The equipment that we work with was this huge spike traps of 2 meters diameter. This is one of the Dermatemis inside of one of those traps. Okay, these are 8 animals, 8 females, and they were attached with the radio transmitters. This is before we did that. And this is a kind of boat with huge antennas that we use. This is a cayuco with the nets and the people who are helping us. This is one sub-adult with the radio transmitter. As you can see, these radio transmitters, they were like very old-fashioned, and they were attached with the silk. Now, the project could use better equipment. This is Paco Soberon that was helping at the field, at the station in Los Tuxtlas. This is Don Miguel Álvarez del Toro, who was the founder of the Natural History Instituto in Tuxtla Gutiérrez, who always helped the project and of Dermatemis. Dr. Rose Mettermeyer from Conservation, when he was in Conservation International, helped a lot to fund the project. And this is our new group of Dermatemis researchers, all in Mexico. People from all over Mexico. For example, this is Gustavo Aguirre from the Institute INECOL in Jalapa, who works with Dermatemis. Dr. Vogt, Kenya, myself, Marco and Judith, who work in Tabasco, Carlos Guichard from the Instituto de Historia Natural in Chiapas, Claudia Centeno, who works in Tabasco and does a lot of field work with her students, and Ricardo Hernandez, Pedro, and other people from Semarnat also. So we hope, we continue working, but we hope we get funds and we can continue working with these really important species. So that's it from my talk, and thank you very much. If you have some questions, I would like to answer them. Sorry.
You have a couple of questions for you.
Yes.
One.
Let me see. Okay. Yes. Where are they?
I can just tell them to you. Can't find
Yeah.
them. Right.
Yes.
What plant species, aquatic and terrestrial, were they consuming?
Oof. They consume a lot of different leaves, but they eat a lot of underwater grass. Also, the Palsatum is the genus of that grass. They also eat mosote, that is like a composite plant with yellow leaves, petals, and brown inside. I mean, I don't know how to say that, but I mean, there are like, those are like the most common, but they can eat fruits, they can eat fruit leaves. For example, a plum, I mean, wild plum leaves they eat, or just the fruit that is falling in the water.
All right, question 2.
Yes.
Without naming locations, are there any robust populations still known to occur in Mexico?
I mean, the most important ones are, there are several, like robust, not very much, but there's some in that area in Montes Azules, but they're like just juvenile. They're not adults, so like the population is not like super healthy. There are some in Centla, in the different rivers in Tabasco, but they're not like big populations. And one population that was very, I mean, surprisingly big was in Rio Hondo, that René Calderon was working with when I worked there a few years ago. I mean, like, not a few years ago, like 10 years ago.
Good deal. That was our last question.
Yes. Okay, thank you very much.
Our next speaker is coming on now, Sarawak in Malaysia. Hello, Siti, are you there? I'll turn it over to you.
Okay, can you hear me?
Absolutely.
Okay, cool. So I'll just go straight to share my screen now. Share screen. Right. Cool. Okay. So good day, everyone. Thank you for staying put. And I'm Siti Norbizura from Institute of Biodiversity and Environmental Conservation, University of Malaysia, Sarawak. And I'm very honored to share you the first assessment, home range movement and habitat use of spiny hill turtle, Hyosmis spinosa in Sarawak, Malaysia. So first, let me just run a very quick through on what do we know about the species. It's a very brief summary of the species. So first of all, we know that there are no concerted research that has been conducted on this target species, specifically in the wild. So most of the information gathered were from captivity and also from incidental observations. And then it is listed as endangered by IUCN, perhaps due to the rarity and difficulty in spotting one, and also because of the high numbers of pet threat and also exotic collections. Now, the systematic relationships and also geographic variations within the species or genus remain unresolved because of these particular reasons, to like rarity and also the unknown provenance of the samples. And then there's one interesting article by Spinks on 2012. So he described the potential of cryptic speciation occur in the species, and it is based on a very large confiscated samples from China. So he ran through some genetic analysis and also look at the morphological variations, and he noticed that there could be at least two main forms, insular and also mainland forms. But then the problem with the results, also back to the same problem, it's unknown provenance of the samples, so it's kind of like hindered the process. Now, the species distribute in Myanmar, Thailand, Peninsular Malaysia, Singapore, and also in Borneo. Okay, so that might be graphic photo and not a very good photo to introduce you to Hyoscmis spinosa, but it's a very critical photo to show you that we really need more on-ground information to create sound and conservation protection for this species. So that one particular specimen is a roadkill one from a protected area, unfortunately. And then we talk about the distribution of the species. And they show versatility in utilizing different type of forests. So they could be found in lowland, they could be found in hilly forests, sometimes in swampy areas, and sometimes in wetlands with flooded vegetation. So they're quite versatile. Now, moving on to the study area of the project. So there's just one sub-adult female trying to climb the hill after submerging the water for a while during midday. Now, the study conducted in Sarawak, Borneo, Malaysia. And in this particular area, at least there are 3 main types of forests: mixed dipterocarp, krangas, and also some mountain forest. And then the elevational range up to 20 meter to 991 meter. And the terrain is quite hilly with steep ridge and at least 5 types of soil constitutions occurring in the area, with red-yellow pozoic soil as the dominant one. Now, for this research, we have a very specific objectives to achieve. The first one, we want to know what factors that might influence the habitat utilization and also movement of Spinosa. And then to be exact, to find that the effect of sexual variation, body size, and also environmental factors. And then the second one, we want to know the critical habitat requirements of the species, as in explore the spatial zones of importance for species life, history, and also survival. Now, the methods used in this study is a radio telemetry, and we use hollow hill, RI2B, and also PD2T in duration of 17 months. So the way we attach the transmitter is through drilling through marginal skewts, and then we use glue to the coastal skewts as flush as it can be. And then for distance lower than 30 meters, less than 30 meters, we use measuring tape, but most of the time we use GPS to track the movement. Now, this is just some photos showing the radio tracking sessions. So we chase them to different type of terrain, sometimes high places, and then sometimes to streams, sometimes to thick forest, and many times to nowhere, where it's very difficult and it's very dangerous for us to go down the hill. So we have to abort the mission. Okay, now let me introduce you to 6 important individuals in this project. So we have 2 males and also 4 females. Body size range from 132 millimeter to 15 millimeter, 5 adults and also 1 sub-adult. So the image illustrates how adult female usually found. So they like to hide under covered microhabitats with only the head visible for the researchers. So it's very tricky to find one. You have to be very, very keen to find them. Now, the distribution of those 6 individuals illustrate in this map. So as you can see, the patterns show it's quite clustered, but with occasionally large strips in certain individuals, and we'll find out why later. Okay, so the home range size in this species is about 1 to 60 hectare through MCP, and then 95% KDE is about 0.44 to 3.45. But then in general, the mean home range in males are greater than females, it's about 32.8 hectare, the mean for males. And we found that there are no significant difference, and it's not correlated, carapace length is not correlated to home range size, and also gender does not affect the home range size of the species. Now, moving on to the movement of the species, we found that distance between observations also greater in males than females, and mean monthly movement also greater in males. Of course, sex-related movements are observed. And although it's quite clustered, they also occasionally do some large trips with two types of patterns. So the first one, when they took the large trips, they would stop by the new area and then stay there like for few days without moving. And then after sometimes like after 7 days, they would start do some small movement, very small movement. And then the second one is right after the large trips, they would move around the places, immediately move around the places, and then they would come back to the former point. And the females of the species, they show infrequent movement with shorter distance compared to males, with minimum of 19 meters over 24 hours period. And they also have both genders also shows inactivity period where they stay inactive, presumably when they are in their respective hideouts, like in the burrows or in the under logs, up to 7 days without movement. Now, moving to sex-related movement, you can see that males show greater distance movement compared to females. And then the greatest distance recorded was 967 meters during August. And mean monthly movement at least 3 to 4-fold larger in males compared to females. And also we notice variation of individual. So this one is particular for females only because we have more individuals of females. So we think that apart from gender, there could be some more factors that could drive to the movement. So microhabitat assessment was conducted for a species, in particular the canopy cover. So out of 398 observations, we found that all of them prefer dense canopy cover. And for canopy cover, we divide to 2 quartiles, so the high quartile and also low quartile. So sometimes males, no, not really males, but sometimes they occasionally use the low quartile of canopy cover, with males the most exploratory sex. So they like the open understory vegetation, and they like to try different type of habitat features compared to females. Now, as I mentioned before, and as we saw from the last photo, so adult female, they like to remain inactive on the high canopy cover compared to adult male and also subadult female. Now, moving on to the microclimatic attributes that affect the movement. So we have like ambient temperature, humidity, and also ground temperature. Now we look at the ambient temperature. So the link of ambient temperature to distance is show positive correlation with female and male. They tend to increase their movement once it reaches certain temperature, 26 to 28. But for subadult, it starts earlier than female and male. While we see no correlation between humidity to distance, but then for ground temperatures, usually the increase of ground temperatures will affect the mobile individuals, but not the case for adult female, because they like to stay inactive for many, many days. Okay, so microhabitat preferences of the species is quite versatile. You can see that they choose buttress, water bodies, fallen logs, inside logs with water, and that one, she stay for 3 days inactive in that particular log. And then dense shrubs, leaf litter. Okay, so this image show you one male hide under the logs. So in general, Hyoscmis spinosa, they're associated with logs. They like to hide under underground holes, and they prefer dense forest with high canopy cover. And occasionally they use formation of rocks, but it's quite rare. And then most of the time they're found in covered microhabitats with preferred ground temperature of 24 to 94.4 degrees. Now for microhabitat use, you can see that they prefer logs the most, with boulders the least. That's probably because of the topography of the study area itself, where such features are quite rare, I mean the boulders. So their preferences, surprisingly not that restricted to water bodies, but then they choose some refuge which provide them the same thing, like they choose logs with water inside. So they don't really choose place close to water bodies, but sometimes they do some large trips to those area. And also we notice sexual variation in terms of utilizing microhabitat, where males prefer open understory vegetation, and we never found them covered under dense leaf litter at least. But for adult female, many times we found that they like to hide under leaf litter, dense leaf litter. Okay, so for the conclusions, this study managed to establish parameters of the species, habitat use, and resources. And also as pioneer studies, we managed to contribute to limited knowledge of the species, habitat use, and also the need for landscape heterogeneity for survival. And then in addition, we found that certain habitat features such as logs, dense canopy cover, and dense leaf litter are very important for their survival. I think that's it. Thank you. Oh, sorry, I forgot. If I could just acknowledge the grants that we received for the research. So we would like to thank NRGS, Conservation Leadership Program, and my research assistants, those that were very committed in helping during day and rainy seasons, and staff of IBEC, UNIMAS, and staff of Sarawak Forestry Corporation, and also veterinarians involved in the study. And for your information, all procedures were approved by UNIMAS Animal Ethics. And that's it. Thank you.
One question for you.
Okay.
Do crocodiles eat Spinosa?
Okay, thank you very much for the questions. So I have no exact answer for that. Do crocodiles eat Spinosa? Because they exist, at least for this population. So they exist in sub-mountain forests, and we found no crocodiles around that area. So I have no answer to that. I wonder why you asked.
And we're going to wait just one more minute to see if we have any more come in from YouTube. And then up next will be Cormac Price in South Africa. Any idea why males and juveniles don't use boulders like the females do?
Okay, any idea why males and juveniles don't use boulders? Okay, so as I mentioned before, we found at least 3 observations where females use boulders, but no observation made where males and juveniles use both, use boulders. So I think for females, they prefer any covered microhabitats that they offer them some security or for their thermoregulation perhaps, but we didn't find males and juveniles use the boulders, so I don't really know why.
All right.
Oh, but if I could just add something, so males and juveniles, they show high mobility compared to females, so that's why they don't really use boulder as much as the females, and especially because of the terrain of the study area itself, such features are quite rare, so we don't really see many observations with boulders. Thank you.
Thank you. And up next is Cormac Price.
Hello, everyone. Let me just try and get share screen. And yeah, Okay. Yeah, can everyone hear me well?
Absolutely.
Great. So I don't know if it was planned by the TSA to start with a South African talk and finish with a South African talk, but I'm very grateful to be here. And people may have gathered based on my accent, I'm not actually South African, originally Irish, but came down here to do my PhD on the sideline side necks. So the two species of side neck terrapin we have in KwaZulu-Natal. So yeah, the two focus species are Pelmedusa galileata and Pelio sinuatus. They're both more or less of least concern on the IUCN red list, least concern on the regional red list too, by base and branch. There is major knowledge gaps though, and in particular with their movement and what they require from their habitats. Reference to Stuart Meakin there from 1983 that they were going to be suggested as a target species for telemetry, almost basically 37 years ago. And whether it was no one was crazy enough or smart enough to attempt to put them as a target species for telemetry until 2016 when we've tried now. So that's the IUCN ranges for both species. In the top left there is sinuatuses. You can see his southern range limit is into the province of KwaZulu-Natal and Mozambique border. And then in your bottom right is the marsh terrapin, which is far more widespread throughout the country.
Or Mac, you popped into presenter mode again.
Oh, have I? Okay, sorry. Is this better?
Perfect.
Ah, okay. Yeah, so they're the current distributions of both species, and in the top right is a live transmitter which we attached to an individual sinuatus at one field site, and in the bottom left is actually a dummy tag on an adult marsh terrapin. We initially put dummy tags at the exact same weight as the transmitters on animals here at the University Animal House, just to see if it altered their behavior at all. And so this dummy tag was placed on this marsh terrapin that was kept at the Animal House pond with 8 other individuals. It was placed on about 2 or 3 months before the first transmitters at field sites were put on. They're attached to the marginal scutes via drilling holes in the marginal scutes and then attaching them with cable ties, because we didn't want to have to recapture the animals afterwards when the batteries on the tags failed. I'll go into further detail on the tags on a next slide. So threat faced by terrapins in South Africa, there's quite a lot. South Africa's population is young and growing. There's a large demand on the country's land for agriculture, but also increasingly a high demand with urbanization. So Durban in KZN is about 4.5 million people population. Between Johannesburg and Pretoria, they've got about 11 million. And all these people require food and all these people require water. And water is a very strained resource in South Africa. So pollution from agriculture and mining, invasive species is also an issue. We have some suitable habitat and suitable climate in South Africa for red-eared sliders, but thankfully there hasn't been any prolific sightings of them. There's some sightings, but we don't know if the populations have actually stuck around and bred successfully here. And of course, climate change. So the northeast of this province, KwaZulu-Natal, suffered more or less the worst drought in the last 50 years in 2016. We've only been really recovering from the drought since 2018. So a couple of the examples of the threats that I was just talking about. In the top left is what should be Neomete pan. Neomete pan should be about 4.5 to 5 kilometers long and about 2 kilometers wide in one of our field sites in Dumo. That's a pollution pipe just north of Durban, coming from an industrial area north of Durban. In the bottom left is the fence of the reserve Ndumo. More kind of Vicky's target species, I suppose, tortoises, have been getting themselves in a lot of trouble with fence lines, particularly electrified fence lines, of walking into the electric fence, not realizing the danger and being shocked. In the bottom right is actually an extension and development of a crocodile farm here in the province. So the crocodile being one of the natural enemies and predators of terrapins. So yeah, that's Neometi pan in March 2016 when we began the field work. So yes, 2016 was the worst drought period Ndumo had in about 50 years. And sadly, because of that, we're finding most terrapins in this sort of condition. So that's a desiccated, dried up, serrated hinge terrapin, Peleosinuatus, or as Africans might say, it's terrapin biltong. Another challenge was hippos. So I'm aware that globally, many terrapin and turtle researchers have to deal with certain species of crocodiles and caimans, like we do with the Nile croc here. But unless you're doing terrapin research in Africa, you don't have to deal with these guys. And I must say, I wouldn't wish having to deal with hippos on my worst enemy. So this is how Neometi pan in Ndumo should have looked in March 2016, but it did bounce back to this. This photo was taken from the same more or less position that the dry photo was taken, but this is March 2018. So after 2 years, it did recover after with some heavy rain, and it's back to a flourishing wetland now. Ndumo was actually a Ramsar site, and it was founded in 1924. So that's how we wanted to find our terrapins, happy and healthy looking, not like the terrapin biltong on the previous slide. So the methods, what we were doing with our sideline side decks, it was transmitters. We also did some morphology, and I know the session is called Terrapins on the Move or Turtles on the Move. Our animals certainly did move, but whether it was voluntary or involuntary is hard to tell. So it was the first attempt in Southern Africa that terrapins were fitted with transmitters. We sampled all the morphology, including a tissue sample for DNA was collected. There was 10 sites throughout the province, but we only used 2 focus sites for the telemetry. In the bottom right corner there, we're fully aware that looking up trees isn't a good place to look for terrapins, but this is a colleague of mine, Dr. Shane McPherson. The transmitters worked by signaling out to relay stations. And the relay stations were put up about 10 meters above the ground along the pan, Neometi pan. And the transmitters then would call in once every 2 hours to the relay stations. And the relay stations would speak to what was called a base station, which had a SIM card inside, and the base station would speak to the laptop. So it was all remote, and it was once every 2 hours. So it's a map of Neometi pan. The pink triangles indicate the relay stations. The blue triangle down the bottom right was the one base station we needed, and the base station spoke to the laptop. So yeah, that's kind of how one of our telemetry sites looked. The telemetry results, so we found bimodal activity patterns for both species. So both species were, I suppose, most active in the early morning and late afternoons. They weren't active midday due to basking, and they also weren't really active at night. There was signs of activity at night, but not much. So the tags also recorded shell temperature, and we also got ambient temperature from nearby weather stations, and the tags obviously recorded activity, which was the locomotive movement, temperature, activity, and signal strength. So with the weather station data, we made correlations on the climatic variables that affected or what didn't affect the terrapins, and we found that ambient temperature affected activity most, followed by rainfall, shown here in these plots, and wind showed no effect. But so ambient temperature showed the largest effect on activity of both species, and rainfall did show a significant effect as well. Looking at this graph, what's most interesting is if you see that the green is the tag temperature of the marsh terrapins at Tala, the telemetry site for marsh terrapins, and the orange line is the ambient temperature. So looking at that for the first, say, 10 hours of a 24-hour period, what was interesting is the marsh terrapins seem to stay 5 degrees warmer than the surrounding weather station temperature, the temperatures that were being recorded at the nearby weather station. How they did this was maybe seeking out microhabitats that were warmer and less exposed in the weather station. But it was interesting to show, based on their activity and their moving around in microhabitats, that they could stay 5 degrees above the average air temperature that was being recorded at the weather station. Sinuatus didn't seem to have that ability, but the field site was in a much kind of warmer eco-region than the marsh terrapin field site. Summary of the findings. So it was the first time, even though it had been suggested, as I said, for 37 years, that terrapins be tagged in South Africa. We got 105 days worth of data from 8 individuals of serrated hinge terrapin and 306 days of data captured from 8 individuals of marsh terrapin. It was 8 each, but 10 tags were put on, 2 tags for both field sites and both species sadly malfunctioned. The tags were ultra high frequency too, which was helpful and novel, rather than just VHF. It meant there was a 2-way communication between the stations and the tags, so long as the tags remained in range. A couple of times we thought we were about to lose animals, so we changed the setting. Instead of calling in once every 2 hours, to call in once every, I think it was 20 seconds, and then we manually went out and tracked to try and find them. As I say, it was diurnal and bimodal activity distribution. Most significant variables were ambient temperature, wind showed no effect. The most interesting finding for marsh terrapin, for me, I found personally, was how it managed to stay 5 degrees warmer in the first few hours. This method it collected a lot of data and remotely, which is very helpful for here, considering in Dumo, the Sinuatus field site was about 6.5 hours drive away. With further development of the equipment, so what we initially wanted was to get space use on triangulation between the stations, but the signal strengths were all too similar. But with further development of the equipment, we can actually get different degrees of space use over time as well. Some of the demographic results, I know this was terrapins on the move, but so in the top is marsh terrapin box plots for their shell length, their shell width, and their body mass. This was for the amount of individuals we caught throughout the province. As you can see, in the darker boxes, which is the reserve we collected most data, Tala, all the animals seem to be heavier and longer, males and females in Tala, than throughout the province, because Tala was a protected reserve. Similar enough results for males and females in a Dumo game reserve for shell length, shell width, and body mass. In the reserve, which was established in 1924, the animals appeared to be heavier, longer, and wider. In the top right photo is marsh terrapin attempting to nest. We only got, it was 2 records of nesting animals from February 2016 to October 2018. There was a huge size class bias throughout the province, with only 11.3% of P. sinuatus animals that were sampled being considered juvenile, 7.8% marsh terrapins being considered juvenile. We weren't encountering many juveniles at all, and we were worried that it's the drought conditions that have made it such a high level of nest failure. So yeah, not many nesting records at all. And the bottom right photo is actually Nile crocodile tooth that broken off, a croc tooth stuck in the carapace of Peleosinuatus. So from the previous talk on crocodiles feeding on terrapins, we know for a fact that Nile crocodile will attempt to, I suppose if they're desperate, Nile crocodile will attempt to eat individuals of both of our species. Showing shell length versus body mass over the growth rate, the different kind of growth rates for both species. So marsh terrapins, it's a smaller animal by nature, but we seems to get heavier quicker. And Peleosinuatus, which takes a little longer, but it goes on because it is the largest hard-shelled species on the continent. The only two species that are bigger are the Zambezi softshell and Nile softshell. My PhD was definitely a team effort. It was, I in particular like to thank for field work Nick Evans and Ewan Genevieve, Celine Hansen and Matthew Burnett, a huge thank you for the help with the stats and the writing. Dr. Shane McPherson for field work too, Mohammed Izzat. I would like to thank the genetics lab of Dr. Sandy Willis Monroe, all the staff and the reserves I worked in, and the sponsors below for equipment. Wireless Wildlife, the Ford Wildlife Foundation, and the National Research Foundation. I think I've gone well over time, but I'm more than happy to take questions at this point. Thank you.
We do have a question, Cormac.
Great.
What small tags would you recommend for small to medium-sized semi-aquatic chelonians?
So our tags were developed and made here in South Africa, and they're more like they're modified fish tags. These tags were inserted into yellowfish. It's a radio telemetry tag. It's not GPS, but it is UHF rather than VHF. So it is a two-way communication between the station and the tag. It's small. We made a plastic protective kind of box over these tags to attach on the marginal scutes, but they weighed, I think it was 14 grams, so that they were small. I would only use the tags we used on medium to large size turtles, unfortunately. We didn't tag anything that was shorter in shell length than I think 20 centimeters for both species.
Thank you. I'm going to wait just another minute or two to see if we have anything else, but I want to remind everyone to be sure to tune in next Thursday at 11 a.m. and for session 5, September 3rd. And be sure to check out poster session 1. We have it up for one more week before poster session 2 comes up. And that was the final question. So thank you, Cormac, and thank you everyone for tuning in today, and we will see you next week.
Thank you.